Okay, well, in the absence of our hosts, and in order to kind of stay on schedule, let me make a few comments, and we can get into the scientific program here. I want to thank all of you for coming today, and also thank the, I think, more than 500 people that are listening to this by webcast. And I think there's more than 60 countries represented, so I think that's really a fantastic thing. There's an old saying in epidemiology that the plural of the word anecdote is not data. And so I'm afraid I've got no data for you today. So I'm going to have to go with an anecdote. So by way of introduction, let me tell you a story that I think bears on the issue of prediction a little bit. A few years back, I got an email from an editor at New England Journal of Medicine, and he was asking whether I would write a review on the genetics of type 1 diabetes. And I'm not normally a reader of the New England Journal of Medicine, and I don't normally like to write reviews, but I could clearly see the political potential of writing a freebie for a journal that's got an impact factor in excess of 50. And so to maximize my political benefits, I also asked my department chair to be a co-author, recognizing that, I'd write the article, he'd make a few desultory comments, and it would go in. So I did that. I wrote the article, sent it in, and I figured, a couple months down the road, I'd be looking at galley proofs and everything would be fantastic. So about 2 months later, I got an email, and the email said, the New England Journal of Medicine regrets to inform you that, we are unable to accept your submission. And it went on to say, we receive many articles, and, we can't publish them all, and so forth. So I thought, well, this is a new experience. It's not that I haven't had papers rejected before, but I couldn't think of a time when I'd had an invited review rejected. So I called the editor and kind of asked him, what's going on here, and he said he had basically he had two major concerns. One was, he said, the paper would be much better with an MD as an author. So that was pretty easy to address. But the other concern was, he said, I read through your entire paper, and you never talk about prediction. And he said, after all, why in the world would you do a GWAS study except to predict? There's no other rationale for doing it. So I had spent 25 years studying genetics of type 1 diabetes, apparently for an inappropriate reason, which was that I was interested in the pathogenesis, and I thought this would give me a window into it. But I think this tells us something about, why people look at GWAS studies and how they respond to them. There's a lot of people that think GWAS studies don't work, haven't worked, and a lot of it has to do with this issue of prediction and whether we are truly covering all of the genetic heritability that's involved in the disorder. And never mind that past linkage studies didn't have much predictive value in the vast majority of people, but they did have predictive value in individuals where they were finding mostly highly penetrant alleles. So I think that's a real challenge going forward for us, not just in humans, but in all species, of trying to take genome-wide genetics data and use it as a prediction tool. And so I think the nice thing about this symposium today is we're doing exactly what that editor told me to do, which is we're going to talk about prediction. And we've got a nice lineup of speakers today. They're going to talk about prediction in a number of different model organisms, and I'm really looking forward to learning about it so that I can be better educated about why I have been doing these GWAS studies, because apparently I wasn't doing them for the right reason. So with that introduction, I would like to turn it over to our hosts and sit back and enjoy the speakers today. Thank you very much.
Thanks, Dr. Cannon. Well, good morning, everyone. Welcome to the first workshop on phenotype prediction using genomic data here at the University of Florida. My name is Marcio Rezende. I'm a PhD here at the University of Florida under the Forest Genomics Lab and one of the organizers of this event. Patricio.
Good morning, everybody, and welcome. I'm Patricio Munoz. I'm an assistant professor in the agronomy department here in the University of Florida. I'm the co-organizer of this workshop. And I just want to let you know, what is the idea and where this workshop is coming from. We have been working with these kind of predictions for a while already, and we were trying to increase the volume of people talking about this in the University of Florida. Now, what triggered finally why we did the workshop, it was in a recent meeting in January actually, where they did kind of this And it was actually really successful. The room was overflowed, and then we actually realized that there was a potential that not only for breeders, but there's a lot of people interested in this area. So Marcio is going to give you some housekeeping stuff and
Yeah, I just would like to acknowledge the sponsors who made this available. So the Genetics and G3 journals, and within the University of Florida, Department of Agronomy, the Genetics Institute, the Plant Molecular and Cell Biology Program, the Horticultural Science Department, and the Institute of Food and Agricultural Science Research, which is the IFRS research. I also like to acknowledge Cornelia Frazier and Ms. Hope Permuter for helping us out with the organization. And just explain briefly, before we move to the first speaker, just explain briefly the idea of the workshop. We thought about doing all the questions at the end in the format of a panel discussion. So I would ask everybody, both in the live streaming as well as here present today, to write down your questions, save to the end, and we'll have all the speakers here sitting now in a round table, and we'll talk about it. The live streaming will end at 12:30, so right after the panel discussion, and then we'll break for lunch. And in the afternoon, we're going to come back, and the speakers will do a quick demonstration about their softwares and how to, just briefly, how their software work in this context of prediction. And for the folks attending through the internet, there is a link there that you can ask questions. It will be directed to the email. I'd like to thank Juan Acosta for helping us out with that, and he's just going to sort through that. With that, I think I would like to invite Patricia to introduce Matthias.
So Matthias has been working extensively in genomic prediction, and not only that, but also in GWAS studies and also in gene expression studies. So you're going to find out. Matthias is going to talk today about the challenges and opportunities that to do this kind of stuff actually in perennial crops, which is actually where we think that there is a great potential. So Matthias, please introduce yourself.
Okay. Thank you. So, all right. Can you hear me? You good? All right. Okay. Thanks everyone for coming. Let's see if this is working. There you go. All right. Thanks Marcio and Patricia for organizing this event. What we have seen over the years since we started working this on genome-wide prediction has been really great interest from a number of people in the community, both at the university but also in other places. And we felt that this workshop was a need because we felt that it would be an opportunity to actually clarify a lot of the things to a number of people about the potential of using this approach. So in talking with Marcia and Patricia about this event, we thought that this first talk was actually a great opportunity to really outline the foundations about genome-wide prediction, so that then the next speakers throughout the morning, they can really explore sort of the needs, the little details about this to a greater extent, and perhaps talk about the application of these technologies in other systems than the one I'm going to talk about. So I'm an associate professor at the School of Forest Resource and Conservation. I'm also a co-director of our breeding co-op that works with lobelei pine as well as slash pine. And working with trees, and especially working with conifers, our biggest limitation is the time that it takes to test any plant in the field, because these trees, of course, take a long time to grow. Now, that's part of the reason why we have become extremely interested in this kind of strategy, because it could potentially allow us to very dramatically cut down the time that it takes us to do breeding. My lab is actually located here at the Florida Genetics Institute, and I'm also faculty in the Plant Molecular and Cell Biology program. Now, I just would like to, before we get started, to thank a few people that have been heavily involved in a lot of the work that has been done in the lab. Obviously, first of all, Marcio, but also Patricio, who's now obviously a faculty. But a lot of the work has really been done with through discussions with a number of people, including Dr. Garrick, who's here today, as well as Gustavo, and also Dr. Marcos Rezende in Brazil. So this is really, because it's such a new type of strategy, such a new type of approach, we and everybody else, I think, can benefit a lot from discussions as to how things should be best done. Okay, and finally, there are a number of sponsors that have been involved in supporting this research. I'd just like to point out that USDA has just funded a couple of proposals in this arena, and one of them will actually support these kinds of workshops over the next 3 years. Okay, so in other words, we have a good potential to actually have these workshops going on for at least the next few years. All right. So as I mentioned to you, I was put in charge to essentially establish or outline the foundation of genome-wide prediction. But I'm going to start by telling you a little bit about tree breeding. So this is what I'm mostly involved with in the lab. And as I was mentioning to you before, tree breeding is a very, very slow process. Okay? Breeders have come up with ways by which they can actually do the crosses and generate seeds actually relatively quickly. Relatively quickly here is 4 years. Okay, so I'm not talking like annual crops where this might take a few months. It takes us 4 years, but it's great. And then also some ways to actually propagate a selected genotype. However, the time that actually takes for you to get the plants in the field and make a selection, in other words, tell whether if that plant is a good genotype or not, is a long, long time. Okay, this is our main bottleneck right now. It's the time that it takes us to test. So the dream of any breeder working with a conifer would be that if we could actually take DNA out of individuals derived from that breeding program and actually through their molecular information, through their genetic markers, use them in a prediction model to determine what is the phenotype of those trees, that would be absolutely fantastic. Okay, we could essentially start cranking through one cycle of breeding and selection every 4 or 5 years. All right. For me to put this into perspective, we have this breeding program going on at NC State, at UF. I was just at NC State, so I don't mean to insult anybody. For 50 years, okay, this breeding program in Lobley and Slashpine has been running for 50 years, and we are only in the 4th cycle of selection. Okay. If you compare that to any annual crop, you would know that they would sometimes do 3 or 4 cycles of breeding and selection within 1 or 2 years, not half a century. So obviously we can benefit a lot from the ability to predict genotypes. But obviously this has a broad range of applications. For example, if you could actually use that molecular information to predict phenotypes and predict the likelihood of someone to develop disease, as well as perhaps, and of course, obviously also in cattle breeding or any other type of application where you could take someone's DNA or a sample's DNA and actually be able to define a priori what that individual is going to look like when it reaches maturity. Okay. Now, what I'm going to do today, I have about 35 minutes left. What I'm going to do is I'm going to start by talking about the principles of genome-wide prediction. Essentially establish the foundation, okay? Essentially, hopefully really set out sort of the groundwork so that we can talk more about this throughout the morning in a lot more detail. I'm going to talk also about accuracy of genome-wide prediction. What determines, what are the factors that impact or that define what will be the accuracy of those prediction models, okay? I'm not going to go into a tremendous amount of detail about the models per se or how they're established, but rather the intent here is really to determine, to re-outline what are the factors that become really important, that are really important in defining the accuracy of that prediction. Okay? Then I'm going to talk a little bit very briefly about our own work, and that's primarily the work from Marcio and Patricio on the application of genome-wide prediction to our pine breeding populations. The types of accuracies that we have been observing across traits and whether if they match the expectations from simulations that have been outlined before. And finally, I'm going to talk a little bit about development and application of what we call, what would be perhaps better models. In other words, models that no longer just account for additive effects, but also start accounting for non-additive effects, and how we are actually very excited about using that information in what we call mate pair location. In other words, the idea that you would now guide your crosses using basically these prediction models. Okay. All right. So how do we start? So, first step in developing these genome-wide prediction models, you need a population that we call typically a training or a discovery population. And I'm working here, I'm just showing you here some trees, but this could be really any type of population. And the intent is to use that set of individuals to really create your model, to really create your predictions, okay? And then if that prediction, if those models work well, if they're accurate, you could potentially use them in the next generations following that population. So obviously, if you want to do that, one thing that you need to have are phenotypes. You need to know what this population looks like in terms of whatever trait you might be interested about, whether if it's a trait related to disease, or whether if it's a trait related to productivity, for example. And you also need to genotype. You need to define molecular markers, genetic variation throughout the genome for those individuals. The place where this becomes actually quite different from the more traditional association genetics types of approaches is that instead of actually trying to fit every individual SNP or every individual polymorphism in a model and try to define what if that polymorphism itself regulates the trait, what we try to do is actually combine all that diversity, all their genetic diversity information, in other words, fit essentially all the SNPs in a prediction model. Okay, so we try to really combine and capture all the information on genetic diversity throughout the genome to explain our phenotype. Now, of course, if you know some statistics, you would think that if you do this, you would probably overfit your model. So you have to take some approaches to actually address that limitation. One of them is, for example, to take your population, your training or your discovery population, take a set of individuals out, develop your prediction model with that subset, and then see how well the subset predicts the individuals that you took out. And you can do that a number of times, and we call this cross-validation, and it's the standard approach that can be used to define how good your prediction model is. All right? But the bottom line is that at the end, after you develop that model, you essentially have a way by which you can take that model to the next generations and use it to predict phenotypes. Okay? Now, going back again a little bit to the principles, what's behind the idea of the prediction models? Let's say we're talking here, we're looking at one individual. That individual has 1, 2, 3, 4, 5 chromosomes. And across these 5 chromosomes, he's got a number of regions, these green boxes here, that contain polymorphisms that regulate your complex trait. Okay? Now, if we're talking about any type of quantitative phenotype, typically the expectation is that they will be controlled by many genes or by many polymorphisms of a relatively small effect. The idea in genome-wide selection is that if you really cover this genome in markers, which are represented here by these red little dots, you will be very likely to have at least the majority of these QTLs, or these regions that contain QTL, that will be in linkage disequilibrium with the marker. Okay? If you have enough markers throughout your genome, you might be able, through these prediction models, really capture the information across all these QTLs. Now, of course, if you're unlucky, you're going to have some of these regions that may not have any markers, and in this case, your model will not predict any of the genetic variants that's associated with this locus. Okay? But that's okay. We expect that there's going to be some limitations here. Now, one thing to bear in mind here is that when I'm talking about a marker and a QTL, I don't imply necessarily that that marker is the causative SNP or the causative locus that creates that quantitative trait loci. It may be simply a marker that is in linkage disequilibrium with the causative polymorphism. Of course, if you're very lucky, you might have actually a marker that is the causative polymorphism, but the expectation that we have is that the vast majority of these markers that we're sampling in the population, they're really in linkage disequilibrium with the marker. Okay? All right. Now, I just described you sort of a series of steps, but the idea here now is that as we get to this prediction model here at the end, we can now take this training population and all generations derived from it And in these generations, essentially use that prediction model to tell within that generation one, across all the individuals, what their phenotype is going to be by just applying the prediction model that we developed in this training population. And the idea is that you could potentially apply this to the first generation, to the second generation, to the third, and more generations. Now, can you apply this to any number of generations? Most likely not. Okay, most likely not, because over time, you're essentially going to have a decay in that association between marker and causative polymorphism. So let's see what I mean by this. Let's say we're looking here at one individual that is derived from a population that has two founders. Okay, one founder essentially I'm representing here by the red section of the genome, and these fragments in green have been inherited by the other founder. And let's say we have 3 QTLs, 3 loci in this chromosome that define the phenotype. Okay? And I'm representing by these arrows here the markers we have. You can see that we have one marker here that is in linkage disequilibrium with our QTL, another one here that's in linkage disequilibrium, another one here that's in linkage disequilibrium with the QTL. As we move this generation forward, as we cross individuals in the training or discovery population, what's your expectation? Your expectation is that you're going to start having recombination events that will now break that association between marker and QTL. Okay? And as you take this forward more and more generations, what's going to happen is that you're going to have more and more cases where that polymorphism is no longer associated with the quantitative trait locus. All right? Now, one way to address this limitation, of course, is that you could simply put more markers, right? The more markers you would have within your population, the more likely it will be that even though we may have lost this marker here, it's no longer associated with a QTL, we still have one marker that was identified before and associated with a QTL that still captures some of the information from that QTL in our prediction model. Okay? So this is critical. It's sort of, you have to recognize that there are a number of factors here that are very important. Number of markers that you have, the structure of your population with respect to linkage disequilibrium, numbers of QTLs, and so on. Okay? And this is what I wanted to talk about now over the next 5 minutes or so, which are the different factors that will impact the accuracy of your genome-wide prediction. What are the factors that are really critical to make it work or not work? And this is perhaps the question that we get asked the most is someone comes to us and says, well, I've got this population, it's got these properties. Are my prediction models, can I develop good prediction models? How many markers do I need? So I'm going to go through the main factors here. They've been outlined by Hayes in a paper in 2009, but there's a number of factors that are important, but these are perhaps the 4 that are most relevant. First of all is your level of linkage disequilibrium in the population. I always tell to my students, if it's not clear in your head what linkage disequilibrium is, that's the first step that you need to go back to the books to really understand the principles of genome-wide prediction. It's really a very important concept, especially for this area of research. The other one is the training and discovery population. What are its properties? What numbers of individuals do you have to develop your prediction model? And as we're going to see in a few minutes, it's going to become very evident why that's important. The heritability of the trait. Okay? How much variation is there for the trait that you're trying to predict, and how much of it is genetically controlled relative to the total phenotypic variance? And finally, the number and the effect of the QTLs controlling the trait. So I'm going to go very briefly through each one of these aspects so that we can see how important they are. First of all, linkage disequilibrium in the population. Linkage disequilibrium is essentially a function of the effective population size and the recombination rate between any 2 loci in the genome. Okay? So critical here is in the population that you're working, how many founder individuals, how many individuals actually you have with regards to the effective population size, which obviously is different from the census size. Now, one thing to remember is that there are several factors that impact that effective population size. There are factors that impact the entire genome, like, for example, migration, but there are also factors that impact some very localized regions of the genome, like, for example, selection. and also the mating system is important. But the bottom line is this. When you have a population that has a small effective population size, typically, you can look at that in the equation here, that typically implies in a high level of linkage disequilibrium. When you have a population that has very high effective population size, that typically implies in a low level of LD. Okay? In other words, those blocks of linkage disequilibrium are very small here and very large here. What happens when you throw in markers? Well, let's say the markers are being represented by the blue arrows here. You can see that in this population here, if I'm developing a model, I have a number of markers that are in linkage disequilibrium with my QTL. Okay? In other words, markers that are informative in defining my ability to predict the phenotype. Okay? On the other hand, this population down here, your extent of linkage disequilibrium is very weak, and as a consequence, even though you've got a lot of markers, you may not be able to have markers that actually capture as much as possible of the variation that controls the trait. Now, you have ways to address this limitation, and one way to address this limitation is you might simply just add more markers. Okay? The more markers you have, the more likely you're going to be to actually be capturing information on genetic variation at these loci that control your trait. Okay? So linkage disequilibrium is important, but it's also important to remember that as it's not necessarily a limitation, it's a limitation that the breeder can work with, either by structuring its population in a way that has perhaps a lower effective population size, or by simply adding more markers. Okay? All right. The other thing is the training and discovery population size. Now this sort of makes sense, right? When you're going to develop these prediction models, you want to try to define what is the effect of a given allele at a given locus. Now if you have very few individuals in your population, it will be a lot more difficult for you to actually make an estimate of the effect of these markers than if you had a large population. A larger population will allow you to make much better estimates of the effect of these markers. This is a sort of a piece of a paper that came out from Dario Grahapalli and Marcos Rezende out of Brazil. It's interesting because it looks at different sizes of a training population from 0 to 8,000 individuals. It looks at different scenarios with respect to the number of markers. So here you have got 2 markers per centimorgan, 50 QTLs, 20 markers per centimorgan, 50 QTLs. And here you've got the same thing, except that now we have 100 QTLs in these 2 graphs right here. What the 4 different curves show you here are the expected accuracy of genomic selection or genomic prediction with different population sizes for different effective population sizes. Okay, so you go from 10 to 100 effective population size. And what you can see here, I think what the main message I want to convey here is the following, that typically as you get to about 2,000, 2,500 individuals, in most cases this curve starts to plateau. So this, just in a sort of very generalistic standpoint, for the types of populations that we work with, this is what we have been targeting. Populations in this order of about a couple of thousand individuals or more. Now I'm going to show you some examples later on of how we apply genome-wide prediction, and it has actually worked very well in a training population size of only 900 individuals. Okay, that doesn't imply then that if you don't have 2,000 individuals, it's just not going to work. It's just that your models are probably not going to be as accurate as you could achieve. Okay, finally, heritability. All right, how heritable is the trait that you're working with? Okay, the more your additive variance or your total genetic variance is part of the total phenotypic variance, the more likely, the better your models are going to be. So these are some results of work in the lab where we looked at our ability to predict the phenotype for 17 different traits with very different heritabilities. And you can see very clearly here that as the heritability of the trait goes up, our ability to predict that phenotype goes up also. Okay? So again, heritability is another factor that's very important in addition to the training, to the size of the training population, as well as the level of linkage disequilibrium. And finally, the number of QTLs controlling a trait, as well as the magnitude of the QTLs. For the vast majority of the traits that breeders are interested about, or many, and actually many people in other different fields, those are traits of quantitative nature. In other words, they're complex, controlled by a large number of loci with alleles with a very small effect. Okay? Now, as you would expect, if you have a large number of QTLs, you may not do as good of a job in predicting it compared to if you had very few loci controlling the trait with those being of large effect. Okay, so we have observed that at least for the traits that we have been working on, the accuracies tend to be very good or higher when we have a much simpler genetic architecture compared to a more complex genetic architecture. Okay. All right. So this is what I wanted to cover sort of in the initial part of the talk, in the first 20 minutes of the talk. What are the general principles of genome-wide prediction? And what are the factors that are the main limiting factors in terms of you developing a good prediction model? Okay. Now I'm going to talk a little bit about the work that we have been doing in the lab in this area. And again, this has been work primarily from Patricio and Marcio. We began applying genome-wide prediction about 3 to 4 years ago in a population that we call the C-clones population. This population has a number of properties that make it particularly interesting for these kinds of studies. The first one being that it's got a relatively large size, about 900 individuals, but it's got a relatively small effective population size. Okay, so we've got a relatively large number of individuals, but they are really derived from a relatively restricted number of founding parents. Okay, and that, as we were talking about before, the effective population size defines the LD in your population. Therefore, this population is quite suitable for genome-wide prediction. The other advantage about working with this population is that a very large number of traits have been characterized in those individuals, meaning we can start really looking at the impact of things like heritability, like I showed before, on our ability to predict phenotypes. And finally, that population has also been genotyped. Now, the marker density that we have here, 3 markers per centimorgan. Low-value pine has about 1,200 centimorgans, so this accounts for about 3,600 markers. It's actually a much smaller density than perhaps we would like to have, but actually, as you will see, it already allows us to do a pretty good job in terms of the prediction. The other nice thing about this population is that it's clonally replicated in 4 sites across the southeastern U.S. In other words, these exact same individuals are now in 4 different locations, okay, which allows us to start asking questions. For example, if I develop a prediction model with my population established in this site, can I use that prediction model anywhere else in the southeastern U.S.? And as you will see, as you would expect, G by E, genotype by environment interaction, really comes into effect here and really impacts that ability to use your model across sites. Okay? But anyway, with this information then, on these individuals, the marker data and phenotypic data, we developed these prediction models by fitting all the SNPs. This was work primarily, as I mentioned, from Marcio. And here are just 2 traits that we looked at. One is DBH, diameter of breast height. The other one is height of these trees. And the interesting thing to look at here is that these are the accuracies of traditional breeding using BLUP. These are accuracies of our prediction model. And as I mentioned to you, our prediction model is really suboptimal because it has a low marker density compared to what we would like to have. But still, our prediction model already does a pretty good job in the accuracy compared to the traditional BLUP approach. The most important thing to do, to think about here, is that by applying these prediction models, we can now tell from day one whether if a tree is going to be a good tree or not. We have a good ability to predict the likelihood of that tree being a good genotype. In other words, we can reduce the breeding cycle by almost half, or by actually half or more, and thus the efficiency that we have is significantly increased relative to traditional phenotypic selection. Okay? So even though we're not making the accuracies yet that are as high as those achieved through traditional breeding, just by cutting the cycle of breeding and selection, we can achieve very significant gains relative to traditional breeding. So that was very encouraging, especially because the number of markers represented here is really just a fraction of what we think or what we're targeting really for this population now. This population, we almost tripling now the number of markers that will be characterized. The other thing that we were interested in the early work that we did on genome-wide prediction was how well do the different traits do? Can we actually predict all the different traits the same way? And also how well do the different ways of developing these prediction models perform? Okay? And our general observation through time has been that most of the models perform quite similarly across the different traits, to one exception, to the exception where we have a more simple genetic architecture. Okay? In other words, where we have a few loci with a very large effect. Okay? So for example, these two traits, rust and rust volume, those are two disease-related traits. And here we did see a bump up in the accuracy of our prediction models when we use two types of models that perhaps give a higher value to these loci, to the few loci that have the largest effect. Okay? But in general, really for most of the traits that we looked at, all the different methods of developing these models, they perform quite similarly. But I think we're going to see through the day today, through the morning, some advances that would suggest that perhaps other models might perform better. But just continue on that same topic, I just want to show you here our predictive ability, but now looking at that ability with an increasing number of markers. And just to clarify here, this is actually that number times 10. So this is 1,000, 2,000, 3,000, 4,000. And what you can see is that for diameter, which is typically considered the traditional type of complex trait, probably hundreds or thousands of markers of very small effect. We need about 1,000 markers or more to reach the maximum of our predictive ability. Okay? On the other hand, rust, which is considered to have a much simpler genetic architecture with fewer loci of large effect, what we see here actually is that far fewer numbers of markers actually reach the peak of the accuracy, okay, or the predictive ability. Sort of really showing you how that genetic architecture is a factor that's important in defining the ability to predict the phenotype. So here's another question that we were really interested about, a lot of the breeders were asking us about. Can I develop my prediction model based on a population that I've grown in Palatka, in Central Florida? Can I now use that prediction model to estimate what is the phenotype in that same population, but now if I grow it in North Georgia, for example? Okay? So as I mentioned to you, we had individuals that were in 4 different sites. This is Palatka, Nassau, Cuthbert, and BF Grant. These sites are very different with respect to soil, with respect to climate. And what we did here is that we used the data from each individual site to estimate, to develop the prediction model, and then we asked the question, can I now use the model developed in BF Grant to predict the phenotype in Cuthbert, Nassau, and Palatka? Okay, essentially we're asking whether if we can use these models across the southeastern U.S. What you can see here is that these 2 sites here, they're very close together. They're considered by breeders being the same breeding zone. If you develop your model for Palatka, it works very well also in Nassau. If you develop it in Nassau, it works well in Palatka. Okay? In other words, within that given breeding zone, these models stand pretty well. Okay? Now, let's look at the extreme. Let's look at a model developed based on data from Palatka, and let's see if we can predict the phenotype in BF Grant, in northern central Georgia. What you can see now is that model here has much lower accuracies. Okay? In other words, we see the effect of genotype by environment interaction that's kicking in. Okay? And that's no different from any other breeding program. In any breeding program, breeders know that they have to take into consideration these kinds of effects. Okay? Finally, the last part that I want to cover, I think I have about 5-7 minutes, is the part of developing and applying what we call, what would be perhaps better, what might be better models in our situation. Okay? So first of all, just as a reminder, the phenotype is defined by the genotype plus the environment, of course also the genotype by environment interaction, but let's keep it simple here. Now you can further break down that phenotype, the genotype effect, into additive dominance and epistasis. Okay. Now, when we started developing these prediction models, most of the approaches that were out there were really trying to account for the additive effects that would contribute to a trait. There's a lot of discussion as to whether if dominance effects are important, whether if you should account for them or not, but we have seen a number of evidence in breeding through time that dominance is significant and perhaps other non-additive effects are important, and that we should try to actually model them and include them in our prediction models. So there's much evidence out there that the effects are not purely additive, at least in the types of species that we work. So we thought that if we could start incorporating dominance and other non-additive effects, we could potentially start actually developing models that are more predictive. Okay? In other words, that we can actually improve the accuracy of the models. So here's the great advantage of working with the marker information, is that marker information actually allows us to start partitioning the effects from additive, dominance, and epistatic effects, and use those in the prediction model. So I'm just going to focus here on the details of the results that we have obtained. I think that's the most important part here. But this was actually work of Patricio Munoz primarily. Essentially, he first developed models to explain the variable, the trait height, with just additive effects, additive in dominance, additive dominance and epistasis. And he developed them based only on pedigree information. In other words, on information that we had about the relationship of individuals in the populations. But also he developed models to compare based on marker information. Okay? And the big question here is, how do they perform? Okay? How does my marker information, is it really useful in terms of developing better models? And are models that account for non-additive effects like these ones, do they perform better than models that account only for additive effects? And here's just a very quick look at the results. I'm not going to go into a lot of detail, but the main message here is this. First of all, these ones here that are followed by a PED, that means that the model was developed based on pedigree information, not marker information. And what you can see is that invariably, if you look at the accuracies here, is that invariably the models developed based on markers, they perform better than the models based on pedigree alone. Okay, and I'd say that that's not that surprising, but that was, again, that observation essentially confirms that the markers actually represent better the relationship of individuals in a population. The thing that's really quite striking is that as you start getting towards models that are more complex with respect to having, including non-additive effects, what you see is that there's actually a very significant improvement in the correlation, for example, or the accuracy, for example. In this case, compared to this one, was, well, these two cases here compared to the ones that include only the additive effects. Okay? Not only the accuracy gets better, but also the mean square error, in other words, the amount that we are not being able to actually explain, also decreases very, very dramatically. Okay? So these models are really capturing some component of the genetic variance that is significant, that really contributes significantly in terms of improving our prediction models. Now, why do we care so much about this? Is it just to improve the accuracy of our prediction models? No. We care about this because we would like to also apply this information to what we call mate pair allocation. Mate pair allocation is the idea that you, instead of crossing individuals more or less blindly, you would cross individuals that create the best allelic combination in the progeny. Okay? In other words, you can make a very efficient breeding program, but really just targeting those crosses that are the ones likely to be the most productive. Now, this is again that population I was telling you about. It just shows you the design. We had 32 parents crossed to the same 32 parents. Every time that you've got a box here, that means that a cross was made between, for example, this individual and this individual, and the number here in the middle shows the size of the progeny. Now, if you could start crossing these individuals, you would need to make about 500,000 crosses to do all the possible combinations. Okay? Now, a breeding program, of course, is not going to do that. We would like to target the crosses that are more likely to be successful. But that's a very significant question for us, especially because breeders have to climb about 40 feet up in the air to make their crosses when you work with trees. So they would like to do the fewer possible. Okay? So the idea of mate pair allocation is this, that you could actually take all these individuals in your discovery or training population, let's say we took one individual from this cross and one from this cross, and based on their genetic makeup, try to make a prediction of what their phenotype, the phenotype of the progeny would look like. Okay? Now in this case you need to know about non-additive effects, because if you were interested only in additive effects, you would be essentially averaging the values of the parents. Now if you do that for this cross, you can now look at the same question and another cross and another individual and ask what the phenotype of the progeny would be in that case. And if you do that for all 450,000 possible crosses, you could essentially rank your crosses as to whether they will be the ones most likely to perform well or not. Okay? Now, I'm just going to jump here. I have only a couple of minutes, so I'm just going to jump here to the results of this type of analysis. This is a study that Marcio did on essentially comparing our ability, our prediction of the value of the crosses using only the additive effect and using additive plus dominance effects. What you see here, or maybe you don't see, but you've got about 900 columns and 900 rows. So these are the 900 individuals in your training population, and what you've got here in this heat map is the average mean expected value of the family derived from that cross relative to the mean of the population. Okay, so when you've got a very dark color, it means that that cross is likely to generate a population that has a lower phenotype than the one you desire. If you've got a more lighter color, that cross will generate a family that has better values. But what I want to point out to here is that this heat map essentially shows the same information, except that now we included in that prediction model non-additive, the dominance effect. Okay? And what you can see very clearly is that this heat map looks quite a bit different from this one. In fact, the rank correlation is only about 0.82, meaning that it seems that there is a good value in us actually incorporating those non-additive effects in our prediction of what the outcome of crosses are going to be. Okay? So I'd like to stop with that. I think we want to stay within time. But I just want to finish up with sort of just a perspective of where we see this going. Okay? These training discovery populations, they can really be the foundation for future work. For example, as you have climate change coming in, you may have your crops now being under new types of stress that were not before. So you might be able to actually use that training population, phenotype for those conditions, and define the crosses that will be better able to address these new challenges. Same thing if you suddenly need a new product to be generated from your crosses, or if you have, let's say, changing threats. Okay? So thank you so much. I'm going to pass this now to Marcio and Patricio. I think we're going to leave the questions for the end, right? And again, just a quick view of our sponsors. Okay? Thank you.
Thank you, Matthias. Please write down your questions, and we'll have the chance to ask them at the end of the panel discussion. As Matthias briefly showed, there are different methodologies, different assumptions, different approaches to apply genomic prediction algorithms. So for our second speaker, we invited Dr. Gustavo de los Campos. He has a lot of experience with plants, animals, and recently now with humans. Dr. de los Campos is an assistant professor at the Biostatistics Department of University of Alabama. His research focuses on the development of parametric and non-parametric models and algorithms for the prediction of complex traits using dense molecular markers. Please. Sorry.
Well, thank you very much, Marcio, for the introduction, and a special thanks for both to Marcos and Patricio for the invitation. You will see that there is a great coordination with the presentation that Matthias made, but it's a coordination that happened just by chance, because we didn't talk about the presentation, and so, or maybe some inbreeding, academic inbreeding. What is the computer? But she don't have it there? You have internet there? Yes, I have in another Dropbox. So we'll go with the laptop then. Why is it that I cannot see it? Trying to see. Okay. So now we have it. Okay, so now we have it. So in my presentation, I'm going to be talking about an overview of method for whole genome regression and prediction, and also discussing some of the factors that affect prediction accuracy for whole genome regression as well. Yeah? A little bit. Okay. So the type of data that we have, basically, we assume that, as Matthias was saying, that we have a collection of individuals, n individuals, for which, I'm going to move this down, because I have a return here, for which we have measured some phenotype, denoted here by Y, and also we have measured the genotype at a number of positions. In the genome, typically we are talking about a large number of positions in the genome, and if we have single nucleotide polymorphism, we can recode these letters by counting the number of copies of one of the alleles at each of the positions. So at the end, our data set is going to look something like this. For every individual, we are going to have the phenotype and the associated genotype coded in this way. Our statistical learning problem is that of finding a function that will map from the genotype into the expected breeding value or expected phenotype, depending on the prediction problem of interest. In breeding problems, we focus on breeding values. In personalized medicine, we focus on expected phenotype or risk of disease. So basically, we need to estimate using this data, this function, and this is essentially a regression problem. Basically, we are going to try to estimate this function that is going to map from the genotypic information into predictions by regressing the phenotypes on the genotypes. We are going to regress the phenotypic outcome on the genotype information using some regression function, and there's going to be some error term in our model, and this error term is going to represent the environmental effects, but also all the approximation errors that we make when we specify this function. For instance, if there is imperfect linkages equilibrium between markers and QTLs, the proportion of variance that we are going to be able to explain is going to be smaller than the heritability, because some of the signals that are being generated by the genome we will not be able to capture with the marker panel that we have. If there are a lot of interactions and we use an additive model here, also there are going to be approximation errors that are going to go into the error term. So the error term in these models, in the empirical models, is not only representing environmental effects, but all the errors that we make in specifying our function, either because we don't have the right set of markers or we misspecify the function, the regression function. So when we try to implement this regression, we have two main set of challenges. One relates to the complexity of the genetic mechanism. In complex traits may involve complex interaction between genes and between genes and environmental conditions. And an important genetic problem is how can we specify a function here that will be able to capture most of the signals that are generated by a complex genetic mechanism. What I say is to capture, because sometimes, and we found this in genomic prediction many times, a function that is not the true model still may be able to capture 90% of the signal that is generated by the model. An example of that, and that's part of the debate that Matthias was referring to, is the case of epistasis. If you believe in system biology, you will believe that epistasis is pervasive. But it turns out that sometimes we regress phenotypes or markers using additive effects, using the wrong model, and still we can characterize a large proportion of the variance that is generated by the complex system as using an additive model. It means that a complex system may release a large proportion of its signal as an additive model. Okay? And sometimes we don't have that situation. But this is the first problem. How do we deal with the complexity of the genetic mechanism? And a second problem is more of a statistical nature, and it has to do with how do we deal with the statistical problem that emerge when we have a regression model where the number of predictors and consequently the number of effects that we need to estimate is much larger than the number of data points. And some statisticians, not all, call that the curse of dimensionality. But of course, if you work in genetics, you also will believe that you have a lot of genetic information that cannot be a curse. It must be a blessing. The problem is how to deal with that. And there are, of course, statisticians that talk about the blessing of highly dimensional predictors, like Leo Breimer. So when Mewisen, Hayes, and Goddard proposed this idea of whole genome regression, which is basically an idea that states, okay, we are going to regress our phenotypes on all available markers, they were able to implement this regression because in the last 20 years there have been great advances in the area of what are called shrinkage estimation procedures, penalized regressions or Bayesian regressions, that allows us to perform these large P with a small n regressions. That is, regressions where the number of effects that we need to estimate are much, much larger than the number of data points that we have. So let's illustrate the problem using a linear model. Suppose that we are going to regress our phenotypes on the marker genotypes. We have P marker genotypes using a linear model. So this is the additive effect of marker J. This is the model residual. But the number of effects that we need to estimate is much larger than sample size. That is the number of individuals for which we have collected phenotypes and genotypes. A traditional approach to estimate marker effects will be to use, or effects in a linear model will be to use ordinary least squares. And in the case of ordinary least squares, we obtain the vector containing the estimated effects by minimizing the residual sum of squares. So we obtain estimates in a way that they maximize the goodness of fit of the model to the training data set. And that is what we do in ordinary least squares, and that is also what we do in maximum likelihood. We estimate effects so as to maximize the fitness of the model to the training data set. The problem is that when we do that, and when P is large, actually it doesn't need to be larger than n. When P is relatively large to n, the estimate that we derive using this type of estimators have large sampling variability. It means that if I were able to collect a new sample from the population and re-estimate effects, there's going to be a lot of variability over repeated sampling. And therefore the estimate has high mean square error, they are poor quality, poor statistical properties, and there is risk of overfitting. So one possibility to confront these problems is to use what are called penalized regressions, where basically what we do is to augment this objective function that is based on goodness of fit by adding to the objective function a penalty on model complexity. And so here, J of beta is a measure of model complexity as a function of the market effects. And lambda is a regularization parameter that will control the trade-offs in this objective function between goodness of fit, or lack of fit in this case, and model complexity. If lambda is equal to 0, we are back to ordinary least squares. But as soon as we start to increase lambda, and lambda has always non-negative values, we are going to put more emphasis on model complexity, and we are going to favor simpler models, solutions or estimates that are somehow simpler. For instance, sparse models where we are going to be zeroing out some market effects. And there is a large collection of penalized methods available, and basically they differ on the form of the penalty function that we choose. Here are some examples of the penalized regressions. If we make our penalty function to be the sum of the squares of the regression coefficients, this is the method called ridge regression, and it's a method that was first proposed in 1973, so it has been around for many years. And this type of method is what is going to induce is shrinkage of the estimated effects towards 0. So if we compare the ridge regression estimate with the maximum likelihood or ordinary least square estimates, the estimates that we obtain with ridge regression are going to be shrunk towards 0. And because we shrink towards a fixed point, which is 0, that reduces sampling variability of the estimator. And when P is large, it also gives you estimates that have a smaller mean square error than the maximum likelihood or ordinary least square estimates. Now this approach shrinks the market effects towards 0 as a function of allelic frequency. Not as a function of, it's a type of shrinkage that is allele frequency dependent. And if you standardize your marker genotypes so that each marker has a unit sampling variance by dividing by the square root of 2pq each marker, then the extent of shrinkage that this induces homogeneous across markers. My former advisor called this method, this is the social democrat of the penalized regressions, because it basically tried to split the pizza, the genetic signal, homogeneously across markers. Okay? And that may be good or not, depending on the trait architecture and the data set that you have. So there are other methods like the lasso, which uses a different penalty, the sum of the absolute values of the beta j's. This method was proposed by Tishiran in 1996, and this method will induce a solution where some of the effects may be zero out. So this method will induce variable selection and shrinkage. So you have a trait with large effect QTLs, maybe this method will be better than this one. But if you have an infinitesimal trait, maybe this method will be better. We'll discuss that. It turns out that for every penalized method that we have, there is a Bayesian cousin or counterpart. We can get to exactly the same solutions that we get here from the Bayesian perspective. And in the Bayesian perspective, what controls whether we do shrinkage, variable selection and shrinkage, or some combination of these, is the prior density that we assign to the marker effects. So in the Bayesian model, we are going to treat the marker effects as random variables, and we are going to assign a prior distribution to these random variables. Typically, the mean of the prior distribution will be 0, and that will induce shrinkage of the estimate towards the prior mean, which is 0. But the difference is on the shape of the distributions. And we have what is called the Bayesian alphabet, which is a collection of Bayesian models that essentially differ on the prior density assigned to marker effects. And there is a model called base A, base B, BL, etc. Conceptually, we can group these models into classes. If we use a Gaussian prior for marker effects, we are going to obtain estimates that are equivalent to those of ridge regression. So it's going to induce only shrinkage of effects, and it's going to induce a type of shrinkage that is going to be homogeneous across markers. Then we have another group of priors, which are called the priors from the thick tail family, the scale T, which is the prior using base A, or the double exponential using Bayesian lasso. And these priors will induce a type of shrinkage that is going to be differential across markers. It's going to be size of the effect dependent. So the markers that have large effects, the estimated effect will not be shrunk as much as with the Gaussian. And the markers with a small effect will be pushed towards zero much more strongly. And that happens because the priors from the thick tail family, relative to the Gaussian, they have higher mass at zero. So it's stating that we believe that a large proportion of the markers have a small effect and thicker tails, which induce less shrinkage for the markers that have sizable effects. Now, none of these methods induce variable selection. If we want to induce variable selection, we need to move into priors that are finite mixtures, like the priors using base B or base C, where we are going to have a finite point of mass at zero, stating that we believe that with some probability the marker effect will be equal to zero, and then a flat slab, which may be Gaussian or T, so that if the marker doesn't fall into the zero state, if it goes to a non-zero state effect, the effect of that marker will not be shrunk as much as with the other priors. Now, I made a toy example last night to illustrate this. The concept here is that we have a variety of methods that we can use, and the different methods, because there is no enough information in the data to estimate all marker effects, we need to put a penalty or a prior in order to be able to estimate these marker effects, because the number of effects that we need to estimate is much larger than sample size. Now, as a consequence of the fact of that, there is going to be influences of the prior on inferences. Okay, so I made a small example yesterday with some mice data set from the Wellcome Trust, roughly 2,000 individuals, 10,000 markers. I picked 50 of the markers to be the causal mutations, and I simulated a trait of heritability 1/2, and I fitted using a package, BGLR, that implements many of these models. I fitted two models. I fitted one using a Gaussian prior and one using a base B that will do variable selection and shrinkage. So I get the same data set, but I fit the two different models. And this is what I get. If I use a Gaussian prior, this is the square estimated effect, and these are the markers. You can see that at the position where we had the QTLs, there are some peaks, but what the ridge regression does, or the Gaussian prior in this case in the Bayesian setting, is that whenever there is signal, there are many, many markers around that QTL capturing the signal. Even in this case we had the causal variant in the model, but there are many, many markers capturing the signal that is generated in that region of the genome. And also in other regions of the genome, there are some small effects estimated by the ridge regression. Now, when I use model base B, this is the type of picture that I get. Okay? For every position where I had a QTL, or for most of them, I capture the QTL, but only one marker gets the pizza that is available at that position. Okay? And so you can see that the different methods will give you different solutions, and this is something that we need to learn to live with, because there is no information in the data to estimate all marker effects, only using information from the likelihood. We need to bring the prior into the game in order to be able to obtain this estimate. And depending on the prior that we are going to be choosing, we are going to have different solutions about marker effects. So what model is better will become, whether using this prior or this prior is better, it will become an empirical question that we need to assess. And there has been several studies that have tried to compare the prediction accuracy of different methods. And I'm going to discuss first the case of simulation studies and then empirical evidence. In the last year, there have been several studies comparing methods, and we are going to classify roughly speaking the methods into two classes: the method that does variable selection and shrinkage, in this case base B, versus the ridge regression BLAP, which is a method that does shrinkage only. This is a slide from the dissertation of Valentin Wimmer of the Technical University of Munich, where he did a review of several simulation studies, and what he put here is the relative predictive performance of base B relative to the BLAP model. So numbers above 1 means that base B was performing better than the ridge regression BLAP, and below 1 it means the opposite. but he sorted the results based on this index, which is the number of QTLs relative to sample size. It is the complexity of the trait expressed relative to the size of the training data set. And this is important because if we have a very complex trait, but we have an infinitely large sample size, we will be able to infer all the QTL effects. And what you see is the following. As this index, the number of QTLs relative to the size of the training data set increases, the relative performance of base B relative to a ridge regression blab decreases. And so in situations where you have between this index in between 0.25 to 0.5, we have a similar performance, and for indexes above 0.5, the ridge regression blab was performing better. So what is the message here is that what model is better, it doesn't depend only on the genetic architecture. Our first thinking is, okay, it depends only on genetic architecture. If you have large effect QTLs, you use variable selection. If you have an infinitesimal trait, you use a ridge regression or GBLAB type method. It turns out that it also depends on the sample size that we have available. Okay? What about comparison based on real data? This is a slide that we put together with Mario Calus. It was published this year in a review article in Genetics, where we look at the same question, the relationship between the model, the effect on prediction accuracy of the model, trait architecture, and sample size. This is based on 3 studies that were published with Holstein data. And these 3 studies, which are cited here, they assess the prediction accuracy of 2 methods, a Bayesian model that does differential shrinkage of estimate of effects, base A, versus the GBLAB that does only shrinkage. And they evaluated this for 2 different traits: fat percentage in milk, this is in Holstein's, and protein percentage. And the important thing is that fat percentage is a trait that has some large effect QTLs, while protein resembles more the architecture of the infinitesimal model. So we had 2 different statistical models, 2 different trait architectures, and they did this for 3 different sizes of the training population. So we have the 3 elements here in the figure. Okay? So what do we see? When we look at the small training size, less than 1,000 in this data set, and we look at fat percentage, the Bayesian model that does variable selection performs much better than the method that does shrinkage only. That is expected because fat has some large effect QTLs. When we look at protein percentage, the difference between the 2 models is much, much smaller. And again, this is expected because the GBLAB model should be good for protein because protein has a lot of small effect QTLs. What happened when the authors increase sample size? Well, prediction accuracy increased across models and traits. Actually, sample size is the most limiting factor because we are asking too many questions. We want to estimate 50,000 marker effects in these cases because these were based on the Illumina 50K chip with sample size of less than 3,000. So sample size is the most limiting factor. You increase the sample size, prediction accuracy increases. But interestingly, the differences between the Bayesian model and the GBLAB decrease as we increase sample size. And that happens because the influences of the prior on inferences diminish as we increase sample size. As we increase sample size, the likelihood starts to dominate our inferences, and we sort of free ourselves from the assumptions of the prior. So again, what model is better? Well, it depends on trait architecture, but also depends on sample size. If you have a large enough sample size, most of the model will perform relatively well and similarly. Most of the evidence of comparison of different models in plant and animal breeding has shown that with relatively large sample size, there are not many differences between models in terms of prediction accuracy. But all of these data sets are family data. And when you use family data, you are exploiting not only linkage disequilibrium, but you are also exploiting the co-segregation of the alleles at the marker and at the causal loci within the family. Actually, linkage is contributing to prediction accuracy probably more than linkage disequilibrium. So what happens when we move to a case where we have unrelated individuals? Do we also observe that there are no differences between the methods that do variable selection relative to those that do shrinkage? Well, this is an example of an article recently published in PLOS Genetics where basically we compare selection of markers for prediction of human height using family data and using data from unrelated individuals. So basically what we did is that we choose from an independent GWAS, we ranked our markers based on the p-value from the top marker, to the marker with the largest p-value, and then we fitted a GBLAB model where we use the top 10 markers, top 50, top 100, up to top 100,000 markers. Okay? And we evaluated prediction accuracy in validation data sets, but this is family data. This is within the Framingham data set. So when we are validating here, we're validating, trying to predict, say, phenotype of the offspring using information from the parents. And so what do we see? The R-square in prediction in a validation setting increases monotonically with a number of markers, and then it tends to reach a plateau, very similar to the result that Matthias was presenting for trees. And when you see this pattern, you will conclude that there are no benefits of doing variable selection, because the model that uses all the markers gives very similar prediction accuracy, or actually higher than if you use only the top 50,000 markers. So if you look at this, you will say, okay, there are no benefits of variable selection. But what happens if we go to unrelated individuals? Well, the situation is very different. This is with Geneva. The same methods apply only to situations where the training and the validation samples are not genetically related. And in this case, the only thing that we are able to exploit is linkage disequilibrium in the population. And linkage disequilibrium in humans span over very short regions. And so this is what we see. We see a sharp increase in prediction accuracy when we increase the number of markers, then a small region of plateau, and then a decrease in prediction accuracy. And also, the level of prediction accuracy that we attain, an R-square of 0.10, is 1/3 of the level of prediction accuracy that we attain with family data. So again, we see a case where what model is better? Do we need to do variable selection? Well, it depends on the trait architecture. It depends on sample size, and also it depends on linkage. If you have linkage, if the individuals share long chromosome segments, you don't need to do variable selection, because even markers that are far apart from the QTL are able to capture those signals. But if you want to predict in the context of distantly related individuals or unrelated individuals, then you will need to do variable selection. And this is for human height. That is an extremely complex trait. It's not a trait. If you do this for type 1 or type 2 diabetes, you will see a very different picture. With type 1 diabetes, with a few markers, we are able to get reasonably good prediction accuracy. Type 2 diabetes, to a lesser extent, but it's a much simpler trait to predict than human height. So far, we have focused on parametric models. So we try to implement our regression on markers using a linear function. But there are also non-parametric approaches. So we can try to use non-parametric functions, methods to implement this regression. This was first proposed in the context of animal breeding, the idea of applying whole genome regression using non-parametric methods by Gianola, Fernando, and Estela in 2006. And the idea is the following. Again, we discussed already this. If you believe in what we see from studies from system biology, you will believe that epistasis is pervasive. And if we try to model epistatic interactions by using contrast between markers across loci, the type of interaction, the number of interactions that we need to consider is extremely high. If you have 1,000 markers and you want to construct all possible one-way interactions, you get an extremely large number of contrasts that you need to consider. So for complex traits, it is not feasible to try to model epistatic effects parametrically by introducing all the contrasts in the model explicitly. But it has been shown in the area of scatter plot smoothing and other areas that some non-parametric procedures may capture departures from the linear model implicitly, even though without the need of considering all these contrasts. And this is the main idea, and there have been two methods around. for doing these non-parametric whole genome regressions. One is called reproducing kernel Hilbert spaces regressions, and I will focus on this. And another alternative is the penalized neural network, but I'm not going to present results about neural networks today. Just some empirical studies. This is a study by Cross et al. In Genetics in 2010. This study was conducted using 599 lines of wheat. The trait was grain yield, evaluated in 4 different environments. And the authors compared the prediction accuracy, here correlation in the vertical axis for the pedigree model in the green bar, the model using pedigree and markers with a linear specification for the markers, and finally a model that uses pedigree and markers, but it has a non-parametric component for the markers, a kernel method. And so what they observe is that if we compare the pedigree versus pedigree plus markers, you can see gains in prediction accuracy by bringing the markers into the picture across traits. And if you compare now the non-parametric approach with the linear model, what you see is that for one trait, we see a substantial gain. For environment 2, grain yield environment 2, we don't see any gains, and then some small gains here. So the gains in prediction accuracy, in this case by going from the parametric to the non-parametric method, were from 0 for environment 2 to 12% in environment 1. In the same study, they also present results for maize, for grain yield, and for some flowering traits. And for flowering traits, day to anthesis and day to silking, under 2 conditions, well water or stress conditions, they fit the linear model and the kernel method, and in this case, the opposite happened. The linear model was better than the kernel method for these traits. And these traits, the flowering traits, are well known to be highly additive traits. So it does make sense that if you use a linear model, you will get a better prediction accuracy. The message that I would like to convey is that there is no universal machine. Okay? The kernel methods tend to perform very well. In most of the studies that you see, you will see that the kernel method performs either similar to the linear model or slightly better, but there are also studies where the linear model is better. So there is no universal answer. You can say, I'm going to use the non-parametric and I'm going to get better results always. Again, it becomes an empirical question, and what model is better, it depends on the trait architecture and marker density in this case. The neural networks are appealing, but there are still a lot of computational issues in terms of when we want to implement this neural network with more than 1,000 markers, say. If we want to really do 50,000, computational is very complicated. We have discussed, presented some evidence related to the effect of the model on prediction accuracy. Now I'm going to present some results about effects of marker density, sample size, familiar relationships, and finally touch on multi-environment methods. And there is, so effects of marker density. As Matthias said, a natural question always is when people try to implement this, how many markers do we need? Okay? And it turns out that how many markers you need, it will depend, as Matthias said, on the linkage disequilibrium. If you have long span of linkage disequilibrium with a few markers, you may be able to do very well, but also on linkage. If you have family data, in addition to linkage disequilibrium, you are going to exploit linkage, and that will help. And with a few markers, you will be able to achieve very good predictions. And finally, on sample size, because we may keep on adding markers, but if we don't increase the size of the training data sets, there is a limit of how much we can get out of a final sample. So here are some examples. This is a study from Vasquez et al. Published in Journal of Dairy Science in 2010. And in this study, I think this is the first study where they assess the effect of marker density on prediction accuracy. This is for Holsteins, for net merit. They do this for many traits in the article. And so they fit the model using evenly spaced markers. These are not marker selects on GWAS. These are just evenly spaced markers in the genome. From 0 to 10,000 markers, and they also had a full model that I didn't include here in the graph with 32,000 markers. And what you see is that in this family data, because these Holstein cattle are all closely related, prediction accuracy increases with marker density, but it does it at diminishing marginal rates, and at some point it reaches a plateau, and with 10,000 markers, you are getting levels of prediction accuracy similar to that of the full model with 32,000 markers. So you will conclude that you don't need more than 10,000 markers, and probably with 6,000 you will do okay. But it is not clear why we are reaching this plateau. Are we reaching this plateau because with 10,000 markers, given the span of linkage disequilibrium, we are able to capture all the signal that is being generated in the genome? Or are we reaching this plateau simply because with a final sample size, we keep on adding markers, but we saturate them all? There is no additional information that we can extract. So in order to assess that, we did a similar study with the Framingham data set for prediction of human height with evenly spaced markers from 0 to 400,000 markers. So this is close to 500,000 markers in the GBLAB model. And when you look, and we did this with different sizes of the training data set. And the patterns are similar. Prediction accuracy, when we have family data, prediction accuracy increases monotonically, but then it reaches a plateau. And the level at which we reach the plateau depends a lot on sample size. So as you can see that here, it's not only that we are it's not that we are capturing all the signals that are being generated in the genome. Here sample size is playing the role of the limiting factor in the model. And if we increase sample size, we are able to increase prediction accuracy further. And also see that in humans, you need at least 200,000 markers or 150,000 markers, and this is with family data, to reach that plateau. While in Holsteins, you can reach that plateau with 10,000, and maybe in the three populations that Matthias was mentioning, maybe with 5,000 markers, you are already there in terms of prediction accuracy. So again, we see the same situation. And in addition to, so how many markers we need depends on linkage disequilibrium, on linkage, and also on the size of the training data set. So far we have discussed, I have 5 minutes? Yeah, I think we'll be okay. The effect of marker density and of sample size. And in all the cases, we look at the effect of increasing the total sample size. But there are many ways of increasing sample size. You can increase sample size by bringing new close relatives or by bringing distantly relative individuals. These are two very different ways of increasing sample size. And it turns out that most of the prediction accuracy that we capture with the models that we know so far are exploiting mostly linkage, and to some extent linkage disequilibrium, but a large proportion of the accuracy that we obtain depends on linkage. And in that context, what we call the local sample size, that is the number of close relatives that you have available for prediction, becomes more important. This is some results published in Flow Genetics in 2011 for human height by Makowski et al. And so they assessed, here you have prediction accuracy, here marker density, but they calculated the R-square for validation data sets that didn't have any close relative in the training data set, that had only one close relative, 2 or 3 or more. And what you see is that in all cases, prediction accuracy increases with marker density. But the rate of increase here is very small, and here is much, much larger. And at the end, with 400,000 markers, if you didn't have any close relative available for training, the prediction accuracy, the R-square was 11%, but if you had 3 or more close relatives available for training, the R-square was 0.36. So you can see here that what we may call the local sample size, that is, how, if you want to predict the phenotype of this individual, what matters is how many close relatives you have in the training data set. That may be more important than total sample size. Finally, something about multi-train, multi-environment prediction. It is well known that environmental conditions modulate the effect of genes, so we have genetic by environmental interaction. This is the slide that Matthias presented. I just changed the colors here for the 4 evaluation sites, but I think it's a very nice experiment conducted here at University of Florida, because they had 4 evaluation sites aligned on an environmental gradient. And so they were able to say, assess, like Matthias explained, what will be the prediction accuracy, say here, if we fit the marker effects here. Okay? And the results are very clear. Prediction accuracy decreases as you move along the environmental gradient. If you train and try to predict within Palaka, correlation 0.75, but if we try to predict, and if we move along the environmental gradient, in this case, the prediction accuracy decreases, showing that there is a great extent of genetic by environmental interaction. Now, the question is, can we exploit this for genomic prediction? And for that, we need to move all the genomic models that I discussed, which were single train model, into a multi-environment model, okay? Where we are going to fit together the effects of all, the effect of markers in each of the environment and try to borrow information between environments. And you can do that in a standard multi-train, multi-environment model, or you can try to do this using environmental covariates. And nowadays, in addition to have a large volumes of marker data, we also have large volumes of environmental information. You know, we have information systems that can produce massive amount of environmental data that we can eventually put together with the markers in a model. And that's what we try to do in a recent study that a former postdoc that worked with me, Diego Jarquín, conducted. Basically, what he tried to do was to implement a genomic model where we have highly dimensional markers, but also highly dimensional environmental covariates. Temperature, humidity, radiation measured at different times on the cycle of the plant. The challenge here is that if we try to put all possible interactions between markers and environmental covariates, the model becomes infeasible. We cannot implement the model. If you have 50,000 markers and 100 environmental covariates, you have, this is actually wrong, you have 1 million contrast, 5 million contrast that you need to fit in the model. So it is not possible to incorporate all interactions between markers and environmental covariates explicitly in the model. So as an alternative, what we did was to develop a reaction norm, trying to put together the ideas of the reaction norm and the genomic blab. In the reaction norm, you think that the interaction emerges between, from the product of an environmental score and a genetic score. And that the interaction term becomes the product of the two. And it turns out that if you can put very easily this in the context of the GBLAB, because the covariance structure induced by the reaction norm has a very simple form. So you can fit a reaction norm where you have the main effect of the markers, the main effect of the environmental covariates, and the interactions between all markers and environmental covariates in a GBLAB context relatively easily. And so we did an evaluation using wheat data from France, 139 varieties of wheat, evaluated in 340 location by year combinations. And basically this is some of the bottom line results. This is the prediction accuracy for the model that includes the environmental effect, the means of the environment and the main effect of the markers. Correlation 0.42. Then we brought the main effect of the environmental covariates and the correlation increased by a small amount. And finally, we brought the interactions between markers and environmental covariates in a reaction norm model, and we were able to increase prediction accuracy substantially relative to the models based on main effects only. So there is a lot of genetic by environmental interaction. It is challenging to incorporate all this environmental and marker information, but there are methods that we can use to somehow try to model genetic by environmental interaction in this context. So to round up, what are the factors that affect prediction accuracy? We have seen that linkage disequilibrium is a factor, but also relationships play a central role here. Sample size is a very important factor. Marker density, the genetic architecture of the trait and the model that we use. And the important thing is that all these factors interact in many different ways. So that how many markers we need, it depends on LD, depends on linkage, genetic architecture, and on sample size. So this is the main message that I would like to leave. And most of the empirical and actually the simulation and empirical studies that suggest that these factors interact in complex ways. Some final remarks. With family data, genomic prediction can increase prediction accuracy, roughly speaking, between 5 to 25% in the scale of correlation relative to the parental average. However, we still need to find ways to make whole genome regression work more effectively with unrelated individuals. Our ability to capture information coming from linkage disequilibrium for complex traits is still very limited, and there is a long way to go if we want to apply these methods effectively for complex traits and with unrelated individuals. In terms of models, with family data, there are no measured differences across models, and that is because when you have long chromosome segments segregating, you don't need to do variable selection because the markers and the alleles at the causal loci are traveling together due to linkage. However, we saw that there are benefits of using variable selection if we work with data from unrelated individuals. The non-parametric methods, the reproducing kernel Hilbert space method, typically perform well, but there are situations where the linear model performs better, and ultimately whether we use a parametric or non-parametric procedure becomes an empirical question. Sample size is the most limiting factor, typically, but local sample size, that is the number of records that we have within a close neighborhood of genetic similarity, seems to be what matters the most. Marker density, accuracy increase with marker density, but at some point it reaches a plateau. And how many markers we need, it will depend on linkage disequilibrium, and sample size. Multi-environment models, I think this is an area that there is a lot of potential, especially in plant breeding, to further improve our ability to predict complex traits. And in humans as well, because if we can observe correlated phenotypes, you know something about obesity and about cholesterol, and all of a sudden you start to be able to predict the genetic risk of a score much, much better than if you have only genetic information. And before I finish, I would like to acknowledge collaborators at the University of Alabama at Birmingham, Paulino Perez, Ian Clementini, Sara Vasquez, and David Allison, my former advisor at University of Wisconsin, Daniel Gianola, Rohan Fernando of Iowa State University, and collaborators at CIMIT, John Hickey and Jose Crosa. So this is what I have. Financial support also from the NIH and from two private companies, Aviagen and Arvalis. And with that, I'll thank you for your attention. I'll pass on to the next speaker.
So I just want to mention, and I just want to tell you that if you saw Matias' presentation early in the morning, it's really logical to use, genomic-wise selection in perennial crops, when one's generation can take more than a decade. However, for annual crops, for example, maize, that you can do almost 3 rotations in 1 year, or 3 generations in 1 year, in some companies actually, it might not seem that logical. So that's why actually we invite Jeff Endelman to come here and talk about, genomic selection in annual crops and the challenges and opportunity that these methodologies bring. So Jeff is an assistant professor in the horticultural department in the University of Wisconsin, and is working in quantity genetics, breeding, and genomic selection now in potato.
Really appreciate the invitation to be here. My first time to Gainesville, and it's been on my list of places to visit for quite some time. So, and that was a great introduction, because I think, I guess of the speakers, I have worked the most in breeding programs related to annual crops, mostly barley, wheat. In terms of practical experiences, I've also looked at data from maize breeding and actually cassava. I spent about 6 months working on cassava breeding on a Cornell project, which was a good transition to potatoes, because they're also clonally propagated. So I'll be kind of focusing more on the potatoes towards the end of my talk, and the majority of the research I have to tell you about today comes from my postdoctoral training at Cornell. I've only been a new faculty member at Wisconsin for about a month now. So here's an outline of my presentation. You'll see some commonality with earlier themes. So in particular, I'm going to talk a bit about the G-BLUT prediction model and talk about shrinkage estimation, but in a little bit different way than we heard about earlier from Gustavo. I'm going to be talking about shrinkage estimation of a covariance matrix as opposed to shrinkage estimation of marker effects. And then I'm going to leave the model part of it behind and touch on, I think, what are the real challenges for plant breeders and all breeders is that how do you use these types of prediction models to actually improve the genetic gain in the breeding program. That's the challenge we face. And you can see three themes here that I'm going to touch on. One is what I like to call the time accuracy trade-off, which is the fundamental theorem of breeding from my perspective, and it manifests in different ways depending upon what kind of selection scheme you're using. And then to illustrate that basic idea, I have a case study of a set of interconnected biparental families in maize, and then I'll go into even more detail into some economic results related to within family selection for barley. Okay, so I actually just threw this slide in a few minutes ago after seeing Gustavo's talk because I thought it would be useful for some of you who are maybe new to this area to just clarify a bit about what is a G-BLUP prediction model versus an R-BLUP prediction model. I noticed that those words tend to use interchangeably, and I like to actually point out that there's a distinction, that they're not equivalent necessarily. So it's just a one-way equivalency. So we can model our marker effects as with a Gaussian prior or using the penalized regression framework, and those produce the equivalent answer from a mathematical perspective, and that's why that's called an RR-BLUP model. And that's a model where you're modeling marker effects explicitly. The G-BLUP model comes from a shift in perspective. Instead of trying to model the effects of the markers, you model the effects of the lines or the entries in your breeding program. And the G is the big G is the typical symbol for the genetic covariance matrix. So that's a matrix with dimensions equal to the number of lines by the number of lines. And in that case, your model is based on a multivariate normal distribution for those genotypic values or breeding values. And as this kind of Venn diagram shows, you can convert an RR-BLUP type of model into a G-BLUP model, but the converse is not always true. There are, and I'll show you that you can write G-BLUP models that don't have a marker-based analog. And in particular, that's what the shrinkage estimation procedure will get us. So here's a model statement for what a G-BLUP prediction model would look like. This is a typical mixed model framework. For those of you who are familiar with that, we have a set of phenotypes, Y, a set of fixed effects, which can be used to model things like location or year. The breeding values are modeled with this symbol little g, and the assumption is that they follow a multivariate normal distribution, where the covariance matrix, big G, normally would be written in this form. This is, I guess, the typical form if W denotes our marker matrix with dimensions of, with the individuals in the rows and the markers in the columns. Then if I take W times its transpose, I get something which is proportional to this genetic covariance matrix. So that's a typical formulation of a G-BLUP model. But I want to kind of focus here on this formula and point out that this is actually just an approximation. It's not really the genetic covariance matrix, and this was touched on by Gustavo, I think, quite nicely, and Matthias. So in order to really know what the genetic covariance matrix is, we would have to know the genotypes at the causal loci, which we don't ever have. So let's just say hypothetically we did know what those were, W with a tilde over it, then I could say this would be a correct model statement that this is my true genetic covariance. But we have markers, not causal loci, and at least on a conceptual level, I think it's useful to just think about the fact that when we use the marker matrix, that's the W without the tilde, that's just an estimator. So that's what the hat over the big G represents. It's an estimator of what the true G is. And if we had a truly infinitesimal model, which we never really have for any real trait, and if we had an infinite marker density, then conceptually there's this limiting behavior. But as I point out here in this bottom equation, the point is just that there's always some error in our estimate of what the true genetic covariance is. And the goal is to estimate that, to reduce that error. We want to produce estimators that reduce that error. And there's lots of strategies for doing that, and Gustavo was talking about a number of them earlier. The one I'm going to talk about here comes from the perspective of trying to do shrinkage estimation at the level of a covariance matrix. So we can reduce our expected error for this estimator if we shrink the estimate towards some low dimensional target. And this reduction in the error becomes more pronounced as we have smaller marker densities. So if you think about you're trying to estimate the true relationship at some unknown causal loci, if those are a large number of those causal loci and I have only a very small number of markers, then the sampling error is quite large because I'm very incompletely sampling across my genomic loci. And so this shrinkage procedure becomes more helpful in that situation as the number of markers decreases. Now, one very useful target, as it's called, for this shrinkage is the numerator relationship matrix, the one based on a pedigree. So that's the expected value. And there's a lot of use of that in animal breeding and some in plant breeding. In a lot of the situations I've been involved with in practice, we tend to have very shallow pedigrees because there's a lot of new material coming into the plant breeding program. So there's really, speaking of things as being derived from a base population, doesn't make a lot of practical sense. Or the records are simply incomplete. And so what do you do in that case? Well, I pursued as a research direction to look at this question of could we use an identity matrix times a scalar as a low dimensional target? And because that identity matrix has zero covariance between the different individuals in your population, it actually turns out that it can only improve your accuracy of your predicted breeding values. So GEBV is the genomic estimated breeding value. It can only improve that accuracy when you already have some phenotypic data available for the individuals you're trying to predict. And I just wanted to have a whole slide just to capture that idea because I find that it's often not recognized that there's a continuum really of the kinds of information you might have about the individuals you're trying to make predictions about. You may literally have no information about them. In annual crops, it's often the case that we do have some information about the things that we're trying to predict. And what I say is that there tend to be low accuracy phenotypes. So there's this gradual process because every time you create a new breeding line, you start it from a seed, and you have to multiply that seed. And depending upon the biology of that crop, that can take a long time. It can take years before you have enough seed to actually plant large-scale trials that allow you to estimate the phenotype of that candidate with a high accuracy. And meanwhile, all the way, you're accumulating information. The first year you have a small plot data, then you maybe have data from 3 plots, and so on. So there are a lot of practical situations in plant breeding where you're going to have this. And I wanted to just show you the kind of phenomenon that can occur when you combine the prediction with the markers and the low accuracy phenotype. And to do that, I'm going to analyze. I analyzed a historical data set that was published a long time ago. It's remarkable that we can still say anything interesting about this data set because it's been analyzed to death. But it's a barley family that was planted in 9 locations across North America and evaluated for 2 years with only one replicate, one plot basically per location. 145 progeny. Because it's an old data set, there's only 127 markers, which is the weakness of this particular example. But within a biparental family, 127 is not a terribly low number, actually. You can do a decent job of capturing the LD. So the analysis that I did was I took the phenotypes from the first year, and I would build a prediction model, and then I would validate those predictions using the mean performance across all 9 locations in the second year. So the idea here is that I'm going to assume that all 9 locations represent my target population of environments for my breeding program, and I'm trying to make selections that are for high average performance. I'm going to note here that each entry is tested in only one location. I'm going to assume here that I only have enough seed to plant one plot for each given entry, but I can still choose to allocate my plots across multiple locations, even though each entry can only appear in one location. So this would be an unbalanced design. It's 3 locations for my trial, but in each location I have a completely disjoint set of entries from that family. There are no repeated checks in this analysis. If you look at the kind of results you get from this, you see that as a function of the training population size, which is the number of phenotyped entries, there's no effect on the accuracy. So it doesn't matter how many I have in that family because I, in this case, only have the phenotypic data, so they're treated as independent genotypes. And also, you see there's no difference between whether I split my trial across 1, 2, or 3 locations. The 3 different series all fall on top of each other. But here's what it looks like when you throw in the marker data. So if you construct a relationship matrix that allows you to differentiate the fine degrees of relatedness among these full SIB lines, you see that you see that the accuracy is increasing as a function of the training population size. That's one of the hallmarks of a genomic selection model. And in the limit of zero training population size, we don't get zero accuracy, we get the phenotypic accuracy. So this is sort of the analog of what's been well established for predictions when you have no phenotypic data available. Interestingly, you also can see that there is a differentiation depending upon whether I use I'll put all of my lines in one location or if I split them across the 3 different locations. And you can see that the 3 location strategy has higher accuracy. And the reasoning is that the predictions I'm making are now averaged over 3 locations, which is a better sample of my target population of environments of 9 locations than if I only used 1. So even though I have a completely unbalanced design with no repeated checks, the markers actually provide the connectivity. This is not these types of a design you would never do classically in plant breeding because you had no way of connecting the different environments without repeated checks, and the markers now give you that connectivity. So that was just meant as a bit of a digression to kind of to motivate the idea of why you would want to do predictions when you have low accuracy phenotype data. And the preliminary yield trials that are ubiquitous in annual crop production are kind of a good example of that. Let's come back to the shrinkage story now. So we're going to look at the shrinkage estimator, and this diagram tries to illustrate illustrate what's going on. I have a set of phenotypes that I've measured for these prediction candidates. I could do my analysis based on the assumption that they're all independent, so that would be this covariance matrix here. At the other extreme, I could assume that the covariance matrix is given by the sample covariance, right? That's where I use the marker matrix W as is and multiply it by its transpose. The shrinkage estimator interpolates between the assumption of independent and the use of the sample covariance. And where that falls in between those 2 extremes is governed by a parameter that's known as the shrinkage intensity, which varies from 0 to 1. And as I tried to explain when I motivated this, that shrinkage intensity becomes more intense. It goes towards 1. That means more shrinkage as I reduce the number of markers. So this is a situation where I have a couple different data sets, and you can see they have different population sizes from a low of 270 to a high of 3,500, and they all have thousands or 50,000 markers, but in each case I'm going to randomly sample a smaller number of markers to simulate what a low density genotyping platform might look like. And you can see that the amount of shrinkage increases in every case as I get to fewer markers. But there are differences too between the 4 different populations, and the differences correspond to this ratio right here, which is theoretically governs how the amount of shrinkage that you should do is related to this ratio, which is the ratio between the population size and the square of the coefficient of variation of the eigenvalues of the covariance matrix. That's a big mouthful, but basically this column here measures the dispersion of the eigenvalues, or in other words, how structured your population is. You can get a more intuitive feeling for that if you look at this example here. So I compare both a 2-row barley population and a 2 plus 6-row barley population. So for those of you who don't know barley comes in 2 morphological types. One has 2 rows, one has 6 rows on the inflorescence. And they're managed as separate breeding programs. They don't make crosses between them. They have diverged and been bred separately. So you put them together and you get a population that has a very high dispersion value. That value of 9 is quite high. And consequently, when you look at this ratio, it's quite low. And therefore, shrinkage cannot reduce the mean squared estimation error for these kinds of populations that have high levels of population structure. So the implications of this for prediction accuracy, what I've done in this example, this comes from a paper published in G3 last year. I took the maize family and I assumed that the 44,000 SNPs that I had in that corresponded to the true relationship matrix. Generated the breeding values, added some noise to simulate phenotypes with different heritabilities or different accuracies. Right? The accuracy is the square root of the heritability. So I have different phenotypes, and then I make a prediction. I have a GEBV based on this GBLUT model, and my prediction model I can use either the true covariance matrix, which I know because it's a simulation, that's blue. I can use a covariance matrix based on a random sample of 384 SNPs, and then I can either apply this shrinkage procedure or not apply this shrinkage procedure. And you can see that if you compare the red versus the green, the shrinkage estimation procedure leads to a gain in accuracy, and these gains can be substantial. In this case, up to say about 0.05 in accuracy. What's interesting, I think, about this figure is one that you see that the difference in accuracy between the GEBV and the phenotype can actually be negative. So like here, if I have a phenotype which has a high accuracy, and I run that through a mixed model analysis, where my estimator for the covariance matrix is quite poor. So I have a very high accuracy phenotype, but then I force it into a model that doesn't actually describe the true genetic covariance. That corrupts the prediction, and I get lower accuracy by using the GEBV than by using the phenotype itself. So this is a really important phenomenon to be aware of as you start to make predictions for entries that you have some phenotypes as well. You need to be aware of what is the phenotypic accuracy that you're dealing with. This isn't just something you can see in simulation. This is the same phenomenon is apparent if you look at real data. In this paper, I looked at 3 pig traits. Again, the validation is based on some progeny test breeding values. If you use the high density marker chip, you can get the highest accuracy. If you do these 384 SNP chip estimates, the shrinkage in red always beats the without shrinkage in green. So the bottom line is shrinking your estimate of G can improve your prediction accuracy. And if you don't have a relationship matrix available, this strategy is also possible if you have some low accuracy phenotypic data available. All right, so I'm going to switch gears now and leave the models behind and then try and, again, tackle these questions about how can we use these to improve genetic gain in a breeding program. And my focus is really oriented on the kinds of breeding programs that we deal with in annual crops. So one point I like to make when talking about breeding program design is just to recognize that we have to make 2 different kinds of selections in plant breeding. And one is that we have to make selections when we make crosses. So we have to pick the parents, but then we also have to produce varieties. So, I mean, in plant breeding, you don't, generally speaking, get paid to produce parents. That selection improves the gain of your breeding program, but it has no commercial value. The commercial value comes if you can actually produce varieties that people can plant and produce. And as we heard a bit earlier today, that there may be different types of models that you want to use depending upon these 2 selection strategies, because the portion of the genetic variance that is relevant for the parental selection is going to be less than or equal to the total genotypic value, which is what is the commercial value. And some models may be better at capturing those distinctions. Of course, the breeding cycle is shorter than the variety development cycle time, or it can be. And in fact, that's probably, I think, one of the classic failures of a lot of traditional plant breeding programs is that they don't make this distinction. They tend to wait until they already are about to release a variety before they use it as a parent, which is waiting too long. And the reason why I say that is because if you think about the breeding cycle, you need to understand that there's this trade-off between the cycle time and the accuracy. And that really just comes at one level from thinking about what is a typical equation for genetic gain. All right, our genetic gain depends upon the accuracy of our selections, the intensity of that selection for a given level of genetic variation. And if we're looking at genetic gain per unit time, then of course the time of the breeding cycle is there too. And as this figure illustrates, there's this trade-off. And the trade-off you can understand, say, taking a phenotypic selection as an example. Right, the longer you wait to do your phenotyping, to blow up your seed more, to phenotype it over more years, you get a better estimate of the phenotype and the genetic merit of that individual. But then there's that time involved, and the longer you wait, the shorter your genetic gain per unit time. So you've got to ultimately find what is the strategy that maximizes your genetic gain, and it's going to be different depending upon the economics and a lot of particulars, so it's difficult to speak in generalities about that. I'm going to show you an example where I tried to analyze that in particular for a barley breeding program here in a minute. But on this subject of time accuracy trade-off, just to maybe make this, that trade-off exists for genomic selection as well. And I want to just show kind of a case study that tries to get at that was published just a few months ago. It's really a collaboration between Cornell and the University of Hohenheim. Christian Riedelsheimer is the first author. who did a great job. So what we did here was they had a set of 4 maze lines, 1, 2, 3, 4 in this picture, and they made all pairwise crosses except for one. They didn't make this cross between 3 and 4. But they had a set of families with different levels of relatedness, and we wanted to look at how accuracy depended upon relatedness. This has been a recurring theme today. What I really like about this study was because it showed very clearly how it differentiated between whether you're talking about training on full sibs versus half sibs and how that accuracy decreased. And so this figure really shows it all. Let's suppose I'm going to try and predict the progeny of a family A by B, let's say. And one strategy would be to actually phenotype progeny from the same family to predict individuals from that family. And now these accuracies I'm talking about here are predictions in the absence of any phenotypic data. So I have some set of full sibs from this cross that I will phenotype, and I will try and predict the remnant progeny from that cross. And if you look at the accuracy versus size, that's the highest curve on this figure. Okay, well, let's suppose you didn't have full sibs, but you had half sibs from families that involve both the parent A and parent B. So that would be like a cross between A and C, where C is some other line in your breeding program and D is some other line in your breeding program. If you could capture the information from both parents in half sibs, you get this next set of curves. And there's multiple curves because in that diagram there's multiple ways that you could have this kind of a configuration. Then the third series is, let's suppose only A or B were captured in a cross. Again, lower still. And finally, neither A nor B have ever been progeny tested before. That's the lowest accuracy yet. So we have this kind of series of lower accuracies, and that corresponds exactly to the speed at which you can turn the crank on your breeding cycle. So if you think about it, if I could use, say, another family from my breeding program, which is not totally unrelated, but not closely related to my family of interest, that's A by B, if I could actually get high accuracy from that, I could just start to make selections within that family without ever having to have taken the time to do progeny testing. But that has the lowest accuracy. So again, there's this trade-off, and you have to walk that, you have to find that right balance. So what does that mean? How do you determine the right balance? I'm going to show you how I tried to do that analysis for one of these scenarios, and it's this one here. At the time I did this scenario, this is the data I had available. I'm going to ask the question, what's the maximum genetic gain I could expect to get if I were to do genomic selection in the way indicated by here, where I am going to build a model based on some set of full sibs from this cross, and I will use that data to both advance the individuals that I have phenotyped and the individuals I have not phenotyped. So these results are in press right now. You can get it off the Crop Science website if you're interested. It's all about trying to look at how do we quantify genetic gain within a biparental family. And as I just mentioned, there are really two different pools of selection in this kind of a context. You're going to genotype all the progeny from this family, but you're only going to phenotype a subset. And so you have one group that are phenotyped and genotyped, and the second group that are genotyped only. And as the figures show, the accuracies are different in those two scenarios. If you look, for example, say a training population size of 60, that means 60 of the progeny are phenotyped. The accuracy with one plot is about 0.3 if I only have the genotypic data. If I come over here, it's about 0.42. So again, when you combine the phenotypes with the genotypes, you can get higher accuracy. That's part of the message I'm trying to convey. And if you spend more money to phenotype more plots per entry, you can also get higher accuracy. That's the 3 different series you see. So what I've done is these are data from the Barley study. This is not a simulation. This is based on that Barley data set I just mentioned earlier. So the data points are based on the sampling, and then I fit a nonlinear regression. The solid lines are these curve fits, and those fits, then the solid lines can be used to do an economic analysis. So in the economic model, I'm going to express all of my costs relative to the cost of one yield plot. Really, this is the data I'm talking about are for grain yield, and the conclusions are really specific to the question of can I increase, what's the cost of increasing genetic gain for grain yield. So in grain breeding, we typically often factor about $20 for a yield plot. That's a rough number that's used when people do economic analyses. Ultimately, you don't have to pick that number. It's always ratios of these costs that matter, but just in terms of thinking about it, $20, roughly speaking, is what we factor when we look at this at Cornell, and our collaborators internationally. A key cost that determines the genetic gain is, of course, the cost of genotyping and the cost of developing the line. So you have to develop the line, then you have to genotype it. I assumed in this case study that that cost ranged anywhere from about 1/4 of a yield plot to 2 times a yield plot. So in real dollars, that would be a low of $5, which is certainly lower than anyone that we're doing in the public sector, but maybe we'll get there in the near future, to a high of something like $40, which is more typical of what a chip might cost, or a genotyping by sequencing approach. I've assumed that the total budget I can spend is 200 yield plots worth, and I'm going to fix the total number of progeny I'm going to select from that family. And what determines that? Well, ultimately, that's determined by how many you can afford to test in the next cycle of your phenotypic evaluation. Once you've specified those parameters, there's a number of other design variables that have to be optimized over. So it's a multi-dimensional optimization problem in order to find what trial design parameters maximize the genetic gain. And I'm not going to go into how I did the optimization, but here's the results of what that looks like. You can express the optimal strategy. Again, it depends upon this cost and the number selected. So each square in these kinds of phase diagrams corresponds to the optimal strategy at a given cost and a given number of selected progeny that you're gonna pick from that family. And there are 3 figures because to completely specify how you design the trial, you have to specify one, how many progeny are you going to phenotype and genotype? How many plots are you going to allocate per each one of those phenotyped entries? And then how many additional progeny will you genotype but not phenotype? Right? Those are the 3 things that you have to specify in order to completely fix the trial design. And what you see, some of these things some of the trends are intuitive. So for example, as the cost of genotyping decreases, if I'm selecting a large number of individuals, then my strategy is to have a big family that I phenotype. So the blue is a large family size, 160, and I only use 1 plot per phenotyped entry. Conversely, if I'm up in the upper left corner of this region, that means I have a high cost of genotyping, and I'm doing a very intense selection. I'm going to have a smaller family, and I'm going to allocate more money to phenotyping each one of those individuals in the smaller family. What was surprising was just to see this result here, which shows that the yellow here corresponds to a value of 0. So it wasn't until the cost of line development and genotyping was 1/4 of a yield plot, which would be $5 in the kind of nominal dollars that we talk about in grain breeding. Only then did you see that it actually became profitable to genotype remnant seed from that family. Otherwise, for every additional dollar you had available, that money was better spent towards phenotyping a new individual compared to just genotyping it. So that was a surprising result. The last slide here on this case study is to now look at what's our genetic gain from this genomic selection strategy compared to a pure phenotypic selection, because there is an opportunity cost associated with paying for those genome-wide markers, and that differs for different people depending upon the scale of your breeding program and your technologies. But what I've done here is I've independently optimized what the phenotypic selection could do with the same budget I gave the genotypic selection. And as you would expect, if my genotyping cost gets cheaper, I can do better. So that's why these lines all go up and to the left. It also depends upon the actual selection intensity. But the point I want to emphasize here is that if you look at the scale on the y-axis here, this is a value of 1 right here. And here we're talking about only a few percentage points of increase in genetic gain under even optimistic conditions for doing genomic selection in the way I've just described. So the bottom line is that if you think you're going to do better than that, you're not going to do it for yield at least when you build models that are specific to this full SIB configuration. So coming back to this slide initially, this strategy here where you build your model based on the full SIBs has the highest accuracy, but I've just shown you that there's a limitation in terms of what kind of expected genetic gain we'll get. We need to start to consider other strategies, and people, of course, generally are looking at these other kinds of strategies where you use more distant relatives, not the full SIBs. You're going to use half SIBs, either that come from both parents, maybe they're aunt-uncle relationships. In fact, if you think about the breeding, some of the proposed schemes that Rex Bernardo has suggested for maize breeding, they have these type of aunt or uncle relationships between the training and the validation. And I think those are some important things to pursue and to do some economic analyses like I've just done for this one scenario, to do that for these other cases, to find out what the prospects for genetic gain are. Okay, so we're of course trying to do this in potatoes in Wisconsin, and since I'm only a month on the job, we don't have any results yet, but I just wanted to put in a shameless plug here for potatoes if I can. Wisconsin has about 60,000 acres of potato production, and it's the largest value for any horticultural crop in the state and nationally as well. Potatoes are the most valuable horticultural crop in terms of the gross market value. We eat potatoes in many forms in this country, potato chips, French fries, the fresh market, and we do all of those breeding we breed for all those different markets in Wisconsin. This kind of shows you, roughly speaking, what a potato breeding program looks like in Wisconsin, and it's similar for other programs. Of course, there's lots of details here that I don't expect you to capture, but just to give you some idea of the scope, the first time we evaluate a new breeding line in the field, it's as a single hill, and we have traditionally evaluated between 50 and 80,000 of those per year in Wisconsin, and I'd say that 50,000 is a pretty typical number for potato breeding programs in the United States. And over time, we start to multiply that. So we get a single hill, then we have 8 hills for each entry, and then we can go on to these bigger size trials. But this value right here is kind of important, that parent selection has typically happened in maybe field year 6 or 7. So there hasn't been a real strong distinction between the time to the breeding parent, to the breeding value evaluation versus the variety evaluation. And that's one thing that I'll be working on over the next several years, is to try and reduce that cycle time. We can, of course, do that for when we have markers linked to major genes, and we're in the process of doing that for some important disease traits. So simple marker-assisted selection, which really hasn't been the focus of the seminar here today, but that's an important strategy for plant and animal breeding. In terms of the complex traits, potatoes are in some sense similar to animals in the sense that the pedigree records are really important and have been kept very carefully. And so I know one potato breeder in Australia who is exploring the use of estimated breeding values from pedigrees for potato selection. I'm not aware of anyone doing that in the United States, but that's the direction we're going to be heading in the next year. And in terms of the topic for today, trying to understand the economic value of doing genomic selection, once you establish breeding based on the pedigree records, you have the right baseline for thinking about what is the additional gain that you get when you incur the cost of running those genome-wide markers on your breeding lines. And we're going to start to explore that next year by running genome-wide markers on our field year 3 entries. So in order to do all this, I'm going to need some help, and this is a shameless plug for anyone here or listening online, any graduate students or postdocs. Please look at my website, contact me if you're interested. Let me close by acknowledging the people and the places who have made this possible. So most of this work was done while I was a postdoc at Cornell, and with funding from the ARS, the USDA, and also from the Bill and Melinda Gates Foundation, which is a great resource for agricultural development. And I'd like to thank my mentors there at Cornell, Jean-Luc Yamink, Gary Allen, and Mark Sorrells. Thanks for your attention.
For the final topic of the morning section, we thought about discussing the fact that genomic selection was predicted, was published and developed over 10 years ago. The main goal of the use of the markers was to do prediction. Whereas in the case of genome-wide association studies, the main idea is actually doing inference and identify polymorphism and populate p-values. So as we go through, we see that there might be some overlap in the statistical approaches and perhaps some of the objectives. To discuss that, we invited Dr. Dorian Gary from Iowa State University.
Put up a source. Thanks.
Very good. Okay, thanks very much. Thanks for the opportunity to come to Florida and tell you a little bit about my experiences with genomic prediction and GWAS. I have a strange accent because I come from New Zealand originally. My background was in agriculture, both in plants and animals, and my career interest has been all about trying to improve populations. Of course, populations are improved by selection, and selection relies on prediction. So most of my research activities have been directly related to doing prediction. And over the last 5 years or so, much of that prediction has been based on using genomic information to try and characterize the quality of the prediction. So I was asked to talk about unifying genome-wide association studies and genomic prediction. A lot of people seem to think of these as completely different issues, but that's not quite the way I see it. So I thought I'd just start off by telling you what I mean by a genome-wide association study. I think historically we would talk about it as using knowledge of genetic markers distributed across the whole genome to identify QTL associated with variation in performance. So that's what it used to mean, but what's happened today with the increase in next generation sequencing, I think a lot of people doing genome-wide association studies are really trying to go beyond the QTL and actually find the quantitative trait nucleotide if it's from a single nucleotide polymorphism, or the quantitative trait variant if it's from a copy number variant or a deletion or inversion or some other kind of feature. So genomic prediction might sound a little bit different. It's using knowledge of genetic variants distributed across the genome to enhance the accuracy of prediction. And in most cases we want to be greedy, so we would use all of the genetic variants as well as all of the pedigree and all of the performance and anything else we can lay our hands on that might help the prediction. So it's worthwhile just stepping back a little bit and reminding ourselves about some of the history of prediction, and we've heard a bit about this already today. Our mixed model equation, a vector of phenotypes, using Henderson's notation, we have a vector of fixed effects, we have a vector of random effects. These are the breeding values that we're trying to predict, and we have all the leftover pieces we call the residual. And the usual assumption we make would be that the variance of these breeding values U would be some genetic variance, covariance matrix, which historically we modeled as the numerator relationship matrix times some parameter sigma squared G. And I guess the symbol has changed in the conversion of my PowerPoint to a Windows machine. We'd normally make some assumption about the variance of the residuals. The simplest assumption in single trait would be an identity times sigma squared E. And the way it's happened in most of the world is to fit this set of equations that we know of as the mixed model of equations, which were a set of equations invented by a PhD student at Iowa State University immediately after the war, Dr. Henderson. His thesis came out in 1949. He only ever had one academic position, and that was at Cornell. And while he was at Cornell, he tried to publish the use of these equations and was unsuccessful because they had no known properties. And it was only when he took one sabbatic to New Zealand, where a young New Zealander who understood matrix algebra was able to demonstrate, that was Shale Searle, that the estimates of the fixed effects from these equations were the same as the generalized least squares solutions, and that these random effects estimates were best linear unbiased predictions. So the paper describing that came out in 1959, so 10 years after he had published the equations. And that pretty well revolutionized the whole prediction scenario, because prior to that, to do something like generalized least squares, you had to invert the variance covariance matrix of the phenotypes, and here the only inverse we need is the inverse of the relationship matrix. And Dr. Henderson not only invented these equations, but he also invented a way to construct that matrix directly from the pedigree. So pretty well around the world, that's how we have done prediction to improve populations. So genomic prediction, landmark paper in Mewison, Hayes and Goddard. I think if Dr. Henderson had seen that paper, he would have said, I don't understand what all the fuss is about. All they have done is fitted the equations that I talked about in my thesis. The only subtle difference is they've said the incidence matrix will be the marker genotypes and the SNP effects would be what we fit as random. They are just exactly fitting this, this particular mixed effects model, making some assumption about the variance covariance of the markers, and perhaps the most simplest assumption to make is that they have an identity as a variance covariance matrix, so every SNP would have the same variance and they'd have zero covariance multiplied by some parameter lambda. And that would give you estimates of the SNP effects, but for prediction, for selection, we're interested in ranking the individuals themselves. And one of the definitions of a breeding value that never made any sense to me as an undergraduate student was the one from Falconer, which said the breeding value was the sum of the average effects of the alleles, summed over the pair of alleles at a locus, and summed over all the loci that affected the trait. And that never made any sense to me because from the previous equations that we knew worked very well in selection, there didn't seem to be anything in there about traits or loci. So what was interesting about this methodology is it did show that if you got the SNP effects, you could sum them up, and this was giving you the definition of breeding value that Falconer had said. And they showed a number of methods in their paper, but really the only difference that I would see between those methods is the way in which this lambda is parameterized. So one of their models, they say we know what lambda is and it's a constant, and they called that BLUP. Another one, they said lambda is unknown, and furthermore it varies for each marker. And if it varies for each marker, that means for each locus with one estimate of S hat, you somehow have to estimate its variance at the same time. And of course, we're normally uncomfortable estimating the variance of something from a single observation, but one way to bring in information is to use a Bayesian method, bring in some prior, and they called that Bayes A. And the other model, they said, was maybe that some of these S values should actually be zero and to fit a mixture model. So that's genomic prediction. So one question people often ask is how well does genomic prediction work? And I think this is still the best information I've ever seen to demonstrate how well genomic prediction works. It comes from a publication by somebody who was a postdoc with us at the time, Anya Wolk, and it comes from an experiment that one of my colleagues, Jack Decker's, designed to implement genomic prediction in poultry. And this was, in fact, was in layers, and the idea was to completely change the breeding program design in order to get the same rate of gain, the same level of inbreeding, but do it from a much smaller population, elite population, and turn the generations over faster. And what had to happen in order to implement this is, as you've heard this morning, you need a training population. So we genotyped some of the parental generations and then did some genomic prediction. So this is the first data that we got from that experiment. On this axis here is the accuracy of the prediction, correlation between G and G hat. And on this axis here are a whole lot of traits in layers. So this is puncture weight, so a puncture score, so how easy you can damage the egg, yolk weight, albumin height, and so on. The black line I'm showing you there is the accuracy of conventional prediction. And since that goes up as you go from left to right, that should tell you that I've sorted these traits according to their heritability. And the red line there shows you what the accuracy of the genomic predictions were for predicting the next generation based on using the genotypes of the individuals. And what you can see is that for all of these traits, the genomic predictions were more accurate than parent average. So provided your sample size is sufficient, you can do more accurate prediction using genomics than using parent average, and that's one of the reasons why everybody is interested in this technology. But what's unique about this data set, we can show that kind of result in almost all data sets. What's unique about this data set is that all the sires were genotyped and all the dams were genotyped and the offspring were genotyped. So we could actually predict the genotype of an offspring based on the average of its parents' genotypes. And of course that genotypic prediction would ignore the Mendelian sampling. So this purple line I'm showing you here is the parent average genomic prediction using the average genotypes of the parents. So if the two parents were heterozygotes, we're treating the offspring as if it was a heterozygote, when in real life some of them would have been homozygotes for one allele, some would have been homozygotes for the other. And the remarkable thing is that you can see for some traits, like this one here, the genomic prediction was totally capturing the family effects. The difference between these two lines is Mendelian sampling. And the same was true for this. So the lowest heritability trait and the highest heritability trait, the genomic predictions really weren't capturing Mendelian sampling. They were just capturing family effects. And for other traits in the middle, we did quite a good job of predicting family effects. That's not at all what we expected. We expected that if you had a certain accuracy of prediction, that would be kind of equally divided between the parent average pieces, which are half the genetic variance, and the Mendelian sampling, which is half the genetic variance. I'm not going to talk too much more about genomic prediction, but there are two major problems with genomic prediction, two major flaws. One of them is that we can't tell in advance a new trait that you bring to me where it's going to sit on this line. We can't tell if it's one we're going to be able to do a good job of predicting Mendelian sampling or not. And that's a major problem, because with conventional selection, if you collect some phenotypes and estimate the heritability, we can make pretty good predictions about response to selection and how alternative breeding schemes will compare. We can't do that with genomic prediction. The second problem with genomic prediction, which I'm not showing you any data on here, is that the regression of phenotype on genomic prediction, or the regression of real breeding value on genomic prediction, often departs markedly from 1. And typically it's a regression less than 1 rather than more than 1, which means we have a tendency to over-predict. And that's no problem if you're dealing with something like chickens, where all you're interested in is finding me the best selection candidates to use as the parents of the next generation. But if you're trying to market a product and the amount people are prepared to pay for the product has a value proposition based on your genomic prediction, there is a real risk that when they actually see how good the animals are, they turn out to be not quite as good as you thought they were. And that's a major problem. Okay, so there is an equivalent model with genomic relationships. I've just showed you the Mewison, Hayes and Goddard idea of fitting the mixed model equations with SNP effects, but if we apply the mathematical trick of multiplying an equation by 1, but I choose the matrix I as my 1 to multiply my equations by, I haven't changed anything. And if I recognize that M times S is the breeding value, M is just a matrix with the number of copies of an allele, and if S is the substitution effects, then MS is just summing up the number of copies of allele, which of course is the breeding value. So this is exactly the same as fitting this. So this equation up here fitting the SNP effects is exactly the same as this equation here fitting the breeding values, except that in this case the variance of U now would be the variance of MS, and that would be the sum of the outer products of the covariates for each marker weighted by a covariate specific variance. That would be what it is in general. In some circumstances everybody assumes this is a constant, and one of those would be G-BLUP, for instance. Okay, so mixed model equations with genomic relationships. So what we're doing in genomic prediction is whether we fit the marker effects or whether we do it this way, it's kind of an equivalent model. We can move from random regression BLUP to G-BLUP. So we can either directly estimate the effects of the animals just like we used to do, but using the inverse of the genomic variance covariance matrix, or we can do it in the other parameterization, estimate the SNP effects, and then sum them up. So let's get back to GWAS then, enough of genomic prediction. So the classical GWAS, you would fit a model, your vector of phenotypes, you would fit fixed effects for all the nuisance parameters, herd, year, sex, age, whatever, and then you would fit one of the markers and estimate its SNP effect, and you would have a residual, and you would have to somehow account for the multiple testing, particularly if you have millions of these SNPs to estimate. Now, the problem with doing that approach is that most populations have some kind of structure. So the assumption that you might have made that the residuals follow a variance-covariance matrix, which is a scaled identity, is likely to be incorrect. Particularly in animal populations, we have breeds or we have families, we have all kinds of genetic structure, which means that the variance of the residuals from that model would be, in general, some variance-covariance matrix, which is not an identity. So there are various ways to fix that, and one way to fix it would be to estimate, say, principal components of the structure and then fit those principal components as fixed effects. Another way that is probably more used to what we would do in animals would be to fit the polygenic effect. So to fit just an additive polygenic effect and assume that has a variance-covariance of a relationship and just fit your SNP out here, one SNP at a time. So using pedigree-based relationships, we could adjust for the structure, but in actual fact we can do exactly the same thing by adjusting the structure for the markers. And so you could imagine doing it with just some of the markers, or maybe doing it with every marker other than the marker that we were interested in here. So we could do it with this kind of a model, but this kind of a model is just like fitting MS. So this is exactly the model that we're doing for genomic prediction. So whereas some people see GWAS and genomic prediction as two completely different things, to me, when you're doing genomic prediction, you're trying to sum up the average effects of the alleles, and when you're doing GWAS, you're trying to say what the average effects of the alleles are too. So to me, they are the same thing, but they do have some subtle differences. So another way to think of what we're doing, since this is regression, you could think of regressing a breeding value, or if you don't have breeding values, maybe it's phenotypes, on the dosage of the marker. So here I've shown you is the number of B alleles if you use the Illumina AB marker system. So if I've got linkage disequilibrium, then when I'm homozygous for the A allele, most of my individuals are homozygous little q for my QTL. And when I'm homozygous big B, most of my individuals are homozygous for the big Q genotype. And when I'm heterozygous, I might have all three different genotypes represented. So one of the consequences you can see of linkage disequilibrium being incomplete is that when you do this regression, the regression coefficient you would expect to be biased down a little bit depending on the extent of linkage disequilibrium. And if we were to walk along the genome and repeat this exercise at every one of 50,000 different markers, when we come to another marker like marker 2, which has lower linkage disequilibrium with a QTL, now the individuals that are homozygous A represent all three different QTL genotypes, and that slope of the regression is lower than it would be before. So you could imagine we could run this analysis and we could get all these regression coefficients, and we expect them to get steeper when we get around the QTL and get shallower when we get it away from the QTL, and therefore you might think that these technologies have got everything to do with linkage disequilibrium. And it turns out that other people have thought that genomic prediction has everything to do with linkage disequilibrium as well. This is from Mike Goddard and Ben Hayes showing the accuracy of genomic predictions here on this scale based on the size of their training population for different heritabilities, and they produced the rather sobering results that for 1,000 training animals you'd only be able to account for 20% of the genetic variance. For 3,000 training animals you'd only be able to account for a third of the genetic variance. So you need a very big training population, and that's for a high heritability trait. I've showed it here for the green line here, which corresponds to a heritability of 0.8. And we don't really have any traits that have a heritability of 0.8, but often what we're doing our regressions on is based on the breeding value from a progeny test. So although the original phenotypic record might have a heritability of only 0.2 or 0.3, the heritability of the progeny test breeding value or its equivalent is more like the 0.8 there. So there's a lot of people that seem to think both genomic prediction and GWAS have got everything to do with LD, but what I'm going to try and show you in the rest of the talk is that I don't actually think it's got anything to do with LD. It's not LD that causes any of these things. It's got everything to do with haplotypes. Okay, so if you run those kind of models, people want to make inference somehow about GWAS, and one obvious way to make inference is to plot the effects of these SNPs. So here I've got genome, this happens to be chromosome 1 in cattle, dairy cattle, and here I've got the absolute value of the SNP effects, since whether the slope is positive or whether the slope is negative is just arbitrary according to the sequence around the primer which determined how Illumina labelled the allele. So if you look at the average absolute value, you can see there are some big effects up here, and some people would like to think that these big effects are the ones that represent the biggest QTL. But these things in the models that we're typically using, these are marginal effects with all of the other effects fitted in the model. So it actually turns out that these effects are not very good ways of representing the QTL at all. So we'll just have a little look at what would happen in that kind of model using simulated data. And I'm not a big fan of simulated data, because with simulated data we always get incredibly good results, and we don't seem to see the variation between traits that we see in real data, even though the simulated genotypes might be the same from one trait to another. But this is simulated data based on real SNP genotypes. So we said if we take some real SNP genotypes and we choose some region here, and I choose one of those SNPs, and I imagine that the SNP alleles represent the dosage of a quantitative trait variant, and I'm going to use that as my phenotype. So my phenotype is the real alleles of a SNP. Okay, so it's as if the heritability was 1. And the candidate markers I'm going to use in my prediction might be just the SNPs that immediately, in the immediate neighbourhood of that real SNP that I'm treating as a quantitative trait variant. So I might use some to the left and some to the right. So say 10 to the left and 10 to the right. And we could do this at random positions along the genome. And what happens when we do that, if the markers that I'm using in my prediction are 20, that's 10 to the left and 10 to the right of the one that I've chosen to be an imaginary QTL, what we see is the average squared correlation for prediction is somewhere about 0.8, but you see that the individual predictions are actually a whole lot more or a whole lot less. And this is a really good demonstration of the fact that the genome is incredibly heterogeneous. So it's very misleading to say that in this effective population, this breed, the LD, and give some single number. Because as you move along the genome, some places will have a lot more LD than that, and some places will have a lot less LD than that. So if this happened to be a QTL, we would have done a really poor job of predicting it, whereas some of these others here, we would have done an almost perfect job of predicting it. And if we to change the size of that window and make that window a little bit bigger, then on average we do a better job of the prediction. So here's what the prediction would look like if we use 20 markers, and here what they look like if we would use 40 markers. So this is in the bovine genome, but we'd see similar things in other livestock. So if somebody could tell me where the QTL was roughly, and somebody could tell me what the QTL genotypes were, we could do a pretty damn good job of coming up with a local prediction equation of that QTL, which we could then apply anywhere. But the trouble is, in real life, I might have a QTL here, and I've chosen that particular SNP, but the candidates for prediction are all the other SNPs on that chromosome, plus in the case of cattle, on the other 29 chromosomes, because the whole point of doing this is that I don't know where they are and I'm trying to find them. So if I repeat this exercise, I'm still using exactly the same SNP as if it was the real QTL with a heritability of 1, so that's my phenotype. But now I'm using the whole genome markers. I'm doing a genomic prediction using the whole genome. But once I get my estimates of all my SNP effects, I'm throwing away all but the ones that were local in the region. And if we were doing a really good job of predicting, of finding these QTL, what I would hope is I get correlations that were pretty similar to the ones that I showed you on the last slide, when the only candidates were the immediately local SNPs. But unfortunately, this is what happens. The accuracy drops way down here to less than 0.4. So although the whole genome regression gives perfect predictions of the individuals that are in the training population, the SNPs that are being informative in the prediction and the training data, 60% of that variation or more is coming from markers a long way away. This is actually a window based on 200 SNPs. So that's about 10 centimorgans, about 5 centimorgans to the left and 5 centimorgans to the right. So less than 40% of the predictive ability is coming from markers within 5 centimorgans either side of the real QTL. So that's one of the reasons why if I do the prediction in a close relative, I still get reasonably good predictions. But my prediction is actually pretty long range. It's much more longer range than I would like. So that's one of the problems we have. So inference from the absolute value of SNP effects is not the greatest for reasons that I've just described. It's also not the greatest because these are marginal values, and if it so happens that my panel has two SNPs that are almost the same on it, then the marginal effects of those would be half what they were if there was only one of the SNPs there. So although I see a big SNP effect up here, somewhere down here where the SNP effects are small, that could be a QTL that's bigger than the one out here, but it just happens that there's a different amount of LD in that genomic region. I have more markers there and the effect is being distributed. So we discovered quite a long time ago that we could get much better predictions of the QTL if, rather than looking at the absolute value of the SNP effects, if we computed the genomic breeding value of a little fragment of the genome. And we started off by doing 5 SNP fragments. So take the 5 SNP markers and take the 5 SNP effects and estimate the breeding value of every one of your animals in the or plants in your population for those 5 SNPs, and then compute the variance over those breeding values and report the variance. And that's what I'm showing you here. And originally we started doing it using a 5 SNP window, and we would move it along 1 SNP at a time. And what you can see is it really cleans up this plot. This is exactly the same trait. Now it's very clear that here is your biggest QTL. Here are the next 3 QTL, which are about the same size. And this is taking into account the frequency of those QTL and predicting how much they contribute to variance, not just the size of the QTL. You could have a big QTL, but if it's almost fixed in the population, it doesn't contribute very much to variance. When we started going to higher density, and we were doing some work in maize where we had over 1 million SNPs, having a 5 SNP window was not about data reduction. I was always taught that statistics was about data reduction, but taking 1,000 animals and having 1 million SNPs and 1 million windows doesn't seem to me to be data reduction. So we've now gone to doing these things on 1 megabase at a time, and at least in the first instance, doing them on 1 megabase, which is round about a centimorgan in livestock populations. So if you do that and you come up, you can generate with a Bayesian method the entire posterior distribution for the variance at each and every 1 megabase region in the genome. You can use that to make inference, and one of the inferences you might want to make is whether this region accounts for more than 0 variance. But that's probably only a sensible hypothesis to be considering if you're dealing with a monogenic or oligogenic trait. If you're dealing with a polygenic trait, then the null hypothesis is probably that there's an infinite number of genes and they're spread across the whole genome. So the inference you're wanting to make is whether this particular window accounts for more than the average amount of variance of each of the windows. In cattle, there's about 2,700 1 megabase windows. So you want to know how often in my Markov chain I'm using for reconstructing the posterior, how often did the sample in that particular window account for more than 1/2,700th of the genetic variance, and that's what this axis here is. So what we find is, say, we find some window where 90% of the iterations in our Markov chain, it accounts for more than the average amount of variance, we'd think that should be strong evidence that there is a QTL here. Well, in actual fact, if you then go to the real data, and this is where we've again used real genotypes but simulated the QTL, if we calculate how often there really was a QTL in that 1 megabase region, you find that for 90% it was actually only about 50% of the time. So you're grossly overestimating how often there is a QTL. But as this probability gets higher and higher, closer to 1, it does get up closer to the line y equals x, which is what we would like. But what we find out happens is when we say there's really good evidence there's a QTL here in the centimorgan, turns out often we're kind of right, but the real QTL was in the next centimorgan, not in this one. And that's not captured by that green line there. So if our test is comparing whether the QTL is in this window or the one immediately upstream or the immediately one downstream, then the red line is what we generate. And if we go to 2 centimorgans to the left and 2 centimorgans to the right, then for high probabilities we're pretty well sitting on the line. So this just demonstrates that the fact that you get a really strong signal in a particular window, or you get a really big signal on a particular SNP, doesn't mean that it's sitting on top of the causal. The causal could be 1, 2 or even 3 megabases away in the kind of populations that we're dealing with. And I see a lot of people trying to do inference where they find the SNPs with big effect, they look for the nearest gene and they say that must be what's involved. That's not the case. The precision is not nearly that much for some reasons I'll try and explain in a moment. So I think the answer for a lot of the things we see in GWAS and in genomic prediction is to do with something that I would refer to as multi-locus LD. So it's got nothing to do with the single locus cell or pairwise locus LD that we think about, and it's got everything to do with haplotype structures. And so to me, if we know about the haplotypes, we can determine what the LD is in a region. But if you tell me about the LD, you can't tell me anything about the haplotypes that are in the region. So although haplotypes and LD are related, it's the haplotypes to me that are the causal thing dictating what's going on, not the LD. So just give you a little example. If I had 2 SNPs and I just look at the haploid sense, the 2 SNPs could have the 0 allele, the 2 SNPs could have the 1 allele, or I could have the alternate versions where I've got a 0 and a 1. So there's 4 haplotypes I could recognize if I had 2 SNPs. And so that's what I've represented here with these 4 different colors. And let's just assume that in a particular position on the genome, those are the only 4 haplotypes. If we were to sequence those regions, there are only 4 haplotypes, and these 2 SNPs perfectly reconcile the haplotypes that exist there. And let's now assume that one of these haplotypes carries the big Q allele and the other 3 haplotypes carries the little Q allele. So that's the architecture of this trait. So of course when we go and phenotype, at least in an animal, they're diploid, and we don't have the complications you sometimes have in plants with polyploidy. So we might have a diplotype that has 2 copies of this first haplotype here, or we might have a diplotype that has 1 copy of the first haplotype and 1 copy of the second haplotype. So it turns out in my population, if I ignore whether these haplotypes are paternal or maternal origin, then there's 10 different haplotypes that we could observe. And if I was to look at the genotype of them at the SNP, there would be 0, 1s or 2s at the first locus, there would be 0, 1s or 2s at the second locus, and their real QTL genotype would be what I have represented at the third locus. So what I would like to think was happening when I did my model, whether it's for GWAS or for genomic prediction, I would like to think that I was fitting a covariate which represented the number of QTL alleles. Because if I did that, the substitution effect would be the QTL substitution effect, and the GWAS signal would be telling me about the size of the QTL. But for these 2 SNPs, even though they perfectly represent the two haplotypes that exist, and they represent the diplotypes that exist. If I do regression on the first SNP, then the red, I'm kind of doing a contrast between the red boxes and these whatever they are, brown boxes and the white boxes, and you can see these red ones include the homozygous QTL and include one of the heterozygotes and one that doesn't carry the QTL at all. So it's quite clear that this particular covariate is not going to be able to pick up the effect of that QTL very well. And exactly the same occurs for the second SNP. It represents a mixture of the various QTL genotypes. Now what the LD is that you would observe between the QTL and the SNPs is totally dictated by the relationship between the SNP alleles and the QTL and how often you see each of these 10 diplotypes. So I could give you different populations that only had these 10 diplotypes and you would observe different LD. And of course the technical definition of an LD is the relationships between the alleles and the founder population. But we don't know what the founder population is, and normally we represent LD by what's happening in the data that we've actually got. And as Gustavo mentioned, that kind of captures both linkage disequilibrium and linkage signal. Okay, so that's why I'm not a big fan of LD, but if I knew about the haplotypes that existed in their frequency, then I know everything about what's going on in that particular region of the genome. So let's just imagine now that we do what everybody is interested in doing, and we do a whole lot of next generation sequence because we think that next generation sequence will be better because it should have the causal mutations on it. So here are my 4 haplotypes I had before. Here are the 2 SNPs that I had before. Here's the real QTL. If I was to sequence this haplotype, those columns I've got there, there's 4 here, there's another 4 here, and there's 6 here. So there's 14 columns. They represent the only way a mutation can be concordant with those 4 haplotypes. So we could have all 4 haplotypes having the 0 allele, or all 4 having the 1 allele, in which case that mutation would be the reference. It's not a SNP. So we're not interested in that. So here we have the 4 SNPs where the 1 allele tags one of the haplotypes, and there's 4 different combinations of those. Here we have the 4 different mutations we could find where the 0 allele tags one of the SNPs, one of the haplotypes. And the other ones, these 2 that I had here originally, and these 4 that I have here, represent mutations where 2 of the haplotypes carry 1 allele and 2 of the haplotypes carry the other allele. And so there's 3 of those, 3 unique ones of those, and the other ones are inverses of them. So for instance, this SNP1 is the opposite of this SNP6. So of the 6 sets, there's really only 3 of them. So you don't have to go away and sequence the genome in this region. I can already tell you that that's all of the things you'll find. Every SNP you'll find will have, or any other kind of mutation is going to line up with one of the ones that I'm showing you there. So that means that if I know that there are 4 haplotypes, and I can see that just from these 2 SNPs, I can actually invent all the features without having to spend a cent on an aluminum machine. Okay, so let's suppose the QTL substitution effect is 10. So big Q adds 5, and those ones which should be little q subtract 5. So my QTL effect on the haplotype is 10. And let's suppose that I had these 7 features available on my genomic prediction analyses. 6 of them represent the SNPs that have 2 alleles and have 1 allele in 2 of the haplotypes. And one of them is the tag allele. That's actually the causal mutation. So what would happen if I did a genomic prediction kind of analysis using that sort of data? Well, the first thing I could do, if I knew these haplotypes, is I could fit the marker haplotypes themselves. So I could construct a marker matrix that represents the dosage of the 4 haplotypes and a residual. I could assume a variance-covariance matrix for the haplotype effects, and a little set of simulated data of 1,000 individuals. These are the estimates I get of the 4 haplotype effects, and you can see if you look at the contrast there, I'm getting round about the 10, which was the true size of the QTL, at least between the big Q and any one of those little Q effects. So what happens when we fit a SNP model instead? Well, if I fit the 6 SNPs that had alleles in 2 of the haplotypes and the causal, this is what I get. So a lot of people would look at these effects and say, well, where's the biggest one? Well, that's the biggest QTL, and here's the next biggest QTL here. This SNP5 has actually got nothing to do with what's going on. In fact, the real QTL, the causal SNP, this is random regression BLUP, is only a tiny bit bigger than this other particular SNP here. So even if you had the causal mutations and you do a GBLUP analysis and you look at the SNP effects, you're not necessarily going to find what's really going on. But what's interesting is, since I know the alleles here that sit in each of the 4 haplotypes, I can take a linear function of them and I can predict the breeding value of the haplotype from those 7 SNP effects. And when I do it, the effects I get of those 4 haplotypes are exactly the same as what I would have got if I fitted the haplotype model. So if I knew the haplotypes and fit the haplotype model, or I don't know them, and I just overwhelm the analysis with all the SNPs that could possibly occur in the region, I actually get exactly the same haplotype effects. So then what happens if I look at reduced models? So if I knock the causal mutation off my panel, and I knock one of the other SNPs, so I just use these 5 SNPs here, I get completely different SNP effects. And if you were ranking those for their absolute value, you'd say number 5 is the big one and number 3 is a big one as well. So what happens when I reconstruct the haplotype effects for them? Well, it turns out I get exactly the same answer again. So this tells me that genomic prediction, when we fit markers together, has actually got nothing to do with pairwise LD. Because the first row I showed you contains the causal, so it contains a SNP in perfect LD with the QTL. The next panel I use doesn't, but they each give exactly the same predictions, right? So we actually don't need LD between a marker and the QTL in order for these methods to work for genomic prediction or for GWAS. What we need is multi-locus LD. And so I'm capturing the fact that this linear combination of SNPs can uniquely identify the 4 haplotypes, and the 4 haplotypes are always in perfect LD with a QTL. So that's why I repeat what I said earlier, which is I think the behavior of these methods have got all things to do with the architecture of haplotypes rather than LD. And the architecture of haplotypes is what gives you the LD that you see, not the other way around. So I can repeat it with all those other combinations of markers and I get all the same values again. And as I pointed out, some of these SNPs are just like the other one, but they've been labelled the other way around. I can get rid of those superfluous ones and I still get all the same answers. So there's a huge family of different marker panels that differ in LD that will all actually give me the same genomic predictions and the same haplotype effects. But in this case, I've done all this with a small lambda. And if you choose the lambda, somehow you've got to know what lambda is, that shrinkage ratio. And generally what happens if you're fitting more SNPs, you shrink them more. So if you actually did this in the normal kind of Bayesian context, you'd get slightly different estimates for each of these models because the amount you shrunk the markers would vary from one to the other. So the results are all about linear dependence and linear independence. A vector is linearly independent if it cannot be written as a linear combination of the others. In k-dimensional space, there cannot be a linearly independent set of more than k vectors, and some of those sets will be orthogonal. So generally we're dealing with markers that are in LD, so they're surrogates of each other, so they're not orthogonal. But you can still have a set whose linear combination uniquely predicts your QTL. You can also have sets that are not capable of uniquely predicting your QTL genotype. So equivalent models, the causal variant does not add any predictive power to any set of SNP markers for which the causal variant is a linear combination of the other markers. Any subset of markers for which the causal variant can be expressed as a linear combination gives the same estimates. In any interval, it does not make sense, at least to me, to fit more linearly independent markers than haplotypes. Because as you increase the number of features, you have a bigger thing to store in memory. And when you're doing a Markov chain, you have more features to fit through the Markov chain. So I would much rather fit a panel that had the minimal number of features that captured the haplotype diversity than use, say, the 29 million SNPs that we know segregate in, say, a dairy breed. So coming back to these windows, I've just shown you that the haplotype values can be reconstructed as a known linear combination of estimated SNP effects, but I could only do that because I knew exactly what the haplotype was. And if I did that, I could get the diplotype values by summing the two haplotypes each diplotype carried. But if I didn't know the haplotypes, I exactly know the genotypes that sit in a haplotype, that sit in the diplotype, even though I don't know their phase. So if I actually construct the breeding value for the diplotype without phase information, I get exactly the same information. And so that's why these windows, there's windows of estimated SNP effects not using haplotypes, are giving us much better inference of the QTL. Because we don't have to know the haplotype, and provided the markers are able to reconstruct the QTL alleles, we should get good estimates. Okay. So what happens in real life? Well, in the software that we've developed, we've been lucky to get USDA funds to develop a number of statistical methods for doing genomic analyses, and we've modified that software so that each time you run an analysis, it automatically prints out the proportion of variance for each of the windows, and it also prints out the whole posterior for them. So in a typical analysis, this is Angus birth weight with 50K panel. Here is the number of SNPs in various 1 megabase windows. On average, there's about 20 on the 50K product. This is typical of what I see. A few regions that account individually for more than 1% of the variance, and they fit here on chromosomes 7, 20, 13 and 26. And what's really compelling about this is if I repeat the analysis in other breeds, so this is Angus, Hereford, Shorthorn, 3 British breeds, Limousin, Simmental, Geltby, those are 3 continental breeds, so these are very, very distant breeds. I find the same windows, like this one on chromosome 6, turns up in British breeds, but it also turns up in continental breeds. This one here actually turns up in dairy breeds. We actually know the causal mutation in dairy cattle, a gene called PLAG1. And this region actually also turns up as a huge region even in Bos indicus cattle, which are very divergent from Bos taurus cattle. So it probably means this is a real gene that is able to be manipulated and not impact the survival of the animal. So a gene like myostatin. We have myostatin variants in sheep, in pigs, in cattle and in humans that cause double muscling, but they are different mutations in that same gene. So this one we believe is probably the PLAG1 mutation, but just like I indicated before, in different data sets, the strong signal can appear in the 24th, the 25th, or the 26th window. So just don't assume that the real QTL is sitting right where you are. Now we did notice in this particular breed we got much smaller results than we did in this particular British breed. So we did start looking at other ways to create features, and one of them was a method called BLOSOC, which reconstructs haplotypes based on a phylogenetic tree, and we were collaborating with some people at Aarhus in Denmark who had developed this methodology, and when we applied it to 2 of those regions in Angus, so we just reconstructed haplotypes locally centered on each of the SNPs in this region. We repeated the analysis with the original SNPs and the haplotype. We see the variance of those windows went up quite a lot. So that tells me that the SNPs that we happen to have in that region are not sufficient to create linear functions that resemble the real QTL alleles. And the real QTL allele there appears to have lower frequencies than anything we can pick up with a SNP chip, and when we fit those haplotype effects, we get much bigger effects of the QTL. If we go and use higher density markers like 700K, then the collective variance from the three biggest effects goes from 20% to about 30%. So these do appear to be some quite large QTL, and this is quite remarkable because birth weight and calving ease are incredibly important selection criteria, and they've been subject to selection for a long time. Nobody wants a dead calf from an animal with calving difficulty. If we look at one of these windows, or three of these windows, and actually look at the haplotypes that are actually there, what we find in cattle is at any particular window, if it's got 20 markers in it, then theoretically we could have 1 million different haplotypes. But we don't see anything like 1 million haplotypes. For one thing, we've only got about 1,000 animals, or 2,000 or 3,000 animals in our population. This is what we see here for this window on chromosome 7 with 11 SNPs. Out of 940 animals, so there's only 1,800 haplotypes I've observed, over 1,000 of them are this one here. And just the top few haplotypes represent an awful lot of them. And then there is a big tail with many, many haplotypes that we only see once. And those could be a genotyping error. If I get an error in genotyping any one of those 11 SNPs, that could generate 11 haplotypes I've seen once that are spurious. Or the alternative for these rare haplotypes, these are maternal haplotypes, because in these populations, the moment a bull is used, he's used widely. So any haplotype he had that was rare is immediately not rare in the next generation. But at any generation I will find rare haplotypes which an animal could have got from its mother, and she got from her mother, and she got from her mother. So I'd expect a tail of very, very rare haplotypes, and I'm not going to be able to predict those very well. I showed you the example with 4 haplotypes, so what's really happening is maybe these ones here that I'm seeing quite a few times I can get good estimates for, but the rest of the ones down there I'm going to get very poor estimates for just because I didn't see them very much in my training data. If I look at the haplotypes I see, and these numbers here just represent the decimal representation of a binomial number, it does turn out that we see some of the same haplotypes across breeds, and I got a little bit interested in this until we went further and looked at the frequency of them, and we find actually that this one might be very common in this breed, but we might have only seen it once in one of the other breeds. And part of the problem for that is that some of these breeds are recent importations into North America, and when they were first brought in, they were allowed grading up from the breeds that already existed. So for instance, in Simmental, we might see this particular haplotype because it's actually an Angus founder haplotype that got carried into the population by upbreeding from Angus. Many of the haplotypes we see are not common to all the breeds or are private to one of the breeds. And so that means if I train in one breed, if what I'm really doing when I fit multiple SNPs is estimating a haplotype effect, so I get a haplotype effect in training, as long as my validation or target animal has that same haplotype, I can do a good job of predicting it. But if you bring me an animal that has a haplotype I've never seen before, I get a totally spurious estimate of the effect of that haplotype. So summary, genomic prediction in GWAS can be undertaken in the same analysis. The signal strength depends not only on the QTL but on the nature and diversity of the haplotypes, which is what dictates LD and not vice versa. Strong GWAS signals can be obtained without the causal variant. So to me, it doesn't seem to be a sensible thing to do next generation sequencing to get all 30 million variants, to impute them all on and then try and work out where the big ones are. It will be much more sensible to characterize the haplotypes you've got and then look at the sequence of individuals for the haplotypes you've got, and then with some bioinformatics determine what might be the causal mutations. The idea of imputing 30 million variants and then grinding your way through a GWAS or a genomic prediction just seems to me a good way to use electricity at a high-performance computer center. It does also require that there are not confounding QTL in other regions. So just as I told you, if there are 20 SNPs in a window, we could see 1 million haplotypes. Well, if there are 20 QTL in a trait, just the combinations of those QTL in haplotypes across different chromosomes means there's 1 million possible combinations of those 20 genes. And if my training population only has 2 or 3 or 4,000 animals, I can only see a fraction of the combinations that could possibly be out there. And so that says that many of the QTL alleles, even on different chromosomes, will by accident be confounded together in the data I have. And of course, what we would ideally like is a data set where they're all orthogonal to each other and all equally represented. But to do that, we'd have to have at least 1 million individuals just to characterize what's happening with 20 QTL. And so to look at things like epistasis is going to be really problematic, because to look at epistasis, we need to look at combinations of all those alleles with combinations of the other alleles. And we're just not going to have most of those combinations represented in a small data set we get from training that represents full and half subfamilies. Genomic prediction will be more reliable if the minimum number of useful variants are used for prediction. It will also be much faster to compute. GWAS analyses can help identify and characterize useful variants. So what we are doing at the moment is going to these big regions, finding candidate mutations, and we're populating the next generation of chip with those causal variants. So we are hoping that we'll start migrating from just anonymous markers that are evenly spaced to new chips which have anonymous markers that are evenly spaced and have the actual causal mutations for these large effect QTL. But the bottom line is genomic prediction is an immature technology. It is more accurate than parent average, but it has a lot of problems. The regressions are not one, and we need to find ways to resolve that. It's a fantastic area for graduate students and postdocs to be in, because there is so much virgin territory that is yet to be explored. So thanks very much for the opportunity to share my thoughts with you.
Thanks, Dorian. And please join me to thank all the speakers today, this morning. And so what we're going to be doing now, we're going to invite all the speakers to the front, and actually now is the time to ask the questions if you are gathering your questions around. And we're going to try to create a discussion about the topics that we've been covering today. Let me just tell you that after the panel discussion, we're going to have lunch on the hall here, which is provided actually by the organization and by the sponsors. And that's going to be around 12:30. We are 10 minutes behind, so 12:30, 12:40, right after the panel discussion ends. And we're going to be retaking probably around 1:40, 1:50 in the afternoon, where the speakers that we invite actually are going to be doing some demonstrations of the software they have developed to do this kind of analysis in the case of Gustavo, Jeff, and Dorian. Okay? So I will open the panel of discussions for questions that the audience here on the audience. Questions? So let me just start by I just want to say something that I just want to clarify that there's a lot of people when they start talking about molecular markers and now markers in selection or genomic selection, they believe that this is the magic bullet or the silver bullet that you can do actually whatever you can predict the phenotype. In reality, what we're trying to do here, and we're showing here today, is that we actually were trying to predict the portion of the phenotype that's controlled by genetics. And usually most of the analysis that we work with the additive portion. And we are trying to find we're behind this, trying to predict the whole genomic portion and eventually the whole phenotype. Even when the word should say phenotypic prediction, but actually we could not predict the phenotype using molecular information. And Gustavo today presented some about that, combining the genotypic information and phenotypic information to try to get to that goal. So I would like to open the discussion and tell you guys, tell us a little bit about what do you think about predicting the non-additive effect. We see different opinions here. please tell us a little bit about that. Yeah, we can just pass around.
I don't know if I have a lot more to say on it than what Gustavo presented, but I certainly have done some work with kernels. I mean, Gustavo talked a bit about the RKHS method, which there's also a version of that in the software I developed. And in some data sets, as Gustavo mentioned, you see that you can get an improvement in accuracy over the additive model. And the question is the interpretation of that. Is it capturing? Is it actually capturing non-additive, or is it is the gain in accuracy due to something else? And this, I'll just say, maybe one of the things to bear in mind when you try and answer the question, compare model accuracy, is to ask yourself, what are the data you're using for validation? Certainly in plant, in analyses that have been published on plants, I mean, I don't even, I mean, there may be an example, but I'm not aware of an example where actually pedigree estimated, progeny test estimated breeding values were used for validation, which is the default method for validation in animal breeding, and it's almost always phenotypic means BLUPs or whatever, but they're phenotypes. They're not that are ultimately being used for validation in plant breeding. So that's something also to bear in mind when you try and look at the accuracies, or what are you using for validation, and there may be other interpretations of why one model is better than another.
Okay, well, in livestock, the first comment I would make is that a lot of the time our training data is based on breeding values from a large mixed model analysis. And the definition of breeding value is the sum of the average effective alleles, and the average effective alleles include some of the dominance and epistasis effects based on the gene frequency of those QTL in the population. So a lot of our training data doesn't allow us to fit non-additive effects, even if they were already there, because they've been regressed out in generating the breeding values or de-regressed breeding values that we use as training data. In other data sets where we are using the individual phenotype, yes, I think there is possibilities of finding dominance effects and epistatic effects, provided we're fitting models that directly fit the QTL effects. But I'm not convinced that we're going to do a good job of finding dominance and epistatic effects just from the SNPs that exist. And as I just showed you, the effects of the SNPs we get are not actually reflecting the QTL effect. They're reflecting the linear combinations of those SNPs that predict the QTL times the QTL effect. So you could have a pair of SNPs that does a better job of predicting the QTL when they're used jointly, but that doesn't imply that they are epistatic. When you fit their interaction, you might do a better job. It doesn't imply they're epistatic. But if you're actually fitting the QTL, so if we can characterize the GWAS regions, determine who's segregating the QTL, then we can fit models that directly fit the QTL genotype, or the combinations of QTL genotypes at different loci. We have developed software to fit dominance effects from the SNPs, but it requires fitting twice as many effects. And in simulated data, it does do slightly better jobs of predicting performance, but it's a lot more time consuming because you've got twice as many effects to estimate. So I think the big advantages we're going to make in non-additive effects is on the QTL we've been able to characterize, rather than just blindly applying it to every one of the 50,000 or 700,000 SNPs that happen to be in our model.
I think probably Patricia would be the most adequate to talk here instead of me, because he's led most of that research in the lab. But anyway, the evidence that we had suggests that there is something there that is non-additive and might be dominance, may not be dominance. I don't think we are trying, I think we have essentially to validate this now. This is, from our perspective, this is really going to come into play when we start doing mate pair allocation. This is where we really would like to start exploring these kinds of non-additive relationships. We have a series of experiments now, just actually recently funded by USDA, where we actually trying to look at these mates, these mate allocations that would maximize this dominance effect and that would minimize this dominance effect to see if we really we will see what we predict to see. So I think it's going to take a little bit of time. We're working with pines. But I think we are now at the point where we'd like to see if those predictions that we make, if they really make sense, if what we see as dominance is really dominance or not, and we'll see if we'll be able to do that.
I don't know, I agree with most of the things that have been said. The one important thing to remember, Dorian mentioned that, is that when we talk about the additive model, we are not talking about the genes acting in an additive manner. We are talking about how much of the genetic variance can be captured using an additive model by regression on allele content, and that is what the allele substitution effects explain. So it turns out that highly epistatic models generate a lot of additive variance. So the first problem is how much of the genetic variance cannot be characterized as additive variance. That's the first problem to understand. The second problem is that it turns out that the power that we have to estimate epistatic components is very small. For any given data set, you need to have, this is a very old problem that was well known for pedigree data, that if you want to estimate epistatic effect or interaction effects, you need very special designs where you have very, very close relatives. And the same problem also appears in the genomic context. Maybe not as strong as with the pedigree data, because with the markers we can borrow information between families, but these are, I think, the two things that we need to consider. A, how much variance are we not exploiting with the additive model by fitting an additive model? And B, can we predict the remaining component of genetic variance? Do we have information? Do we have the type of design, experimental design, that is needed to capture these epistatic effects?
Dr. Garrett, just for the live streaming, may you repeat the question, please, just so they...
was about the width of the haplotypes, and I talked about using 1 megabase haplotypes, and should the haplotype sizes be different, or should they be different at different parts of the genome. It's interesting when we present some of our data, you get one of two reactions. A lot of people think instantly the haplotype should be much smaller than that, and that we can get much more precision. We should be fitting like 1/4 megabase blocks, which would be nice, but in the kind of populations we've got with small effective population size, as we saw in the first presentation, the haplotypes can be quite large. So in a population with a small effective population size, the haplotypes are too large. You can imagine you split them into smaller effects, but the rest of the haplotype on either side is going to be confounded with it. So you're really not going to be getting the resolution that you think you're getting. If somebody could tell me a sensible way to resolve what the width should be based on the genomic data, we could change the width along the genome. Our choice of 1 megabase is just an arbitrary choice. If you try to publish the results of these QTL, most journals want a p-value, or they want the posterior plotted. So if you've got to store the samples so you've got the whole posterior distribution, it's a lot of memory if you're going to store smaller regions than 1 megabase. But finally, research is an iterative process. You don't just do one analysis where you try and find everything. So our knee-jerk reaction to initial analysis is 1 megabase windows. If you then hit a window that's of interest, you would then use all kinds of techniques to characterize the haplotypes that are there and fit those haplotypes as fixed effects and so What we've seen in some of the livestock work is that the haplotypes are, can be bigger than a megabase, and other times they can be smaller. The genome is very heterogeneous. So to me, a megabase is not a bad choice just for the first scan of the genome. I think finding the causal variant is going to involve functional genomics and a whole kind of other approaches. And in those approaches, you're obviously not constrained to the particular size that you used in the preliminary analysis. But most people seem to think that the precision they're getting these estimates for, if the density of the panel is 800,000, then they think the precision that we're getting these estimates is very small, and that's just not the case, because the haplotypes that we see in a small effective population size are typically quite big. Humans would be quite different.
So two questions that came online that follows up to Jared's question. The first one is whether Dr. Garrick used tag SNPs in his analysis. I know this.
We're not using tag SNPs, because when we run a routine analysis, we're using the genotypes that come off the Illumina platform. So we don't know what the haplotypes are. So what I was just demonstrating in an example is, if I know what the haplotypes are, I can show you that when I fit a model ignoring them, the model is actually taking account of the relationships between those SNPs that form the haplotype, even in my ignorance of it. But yes, if somebody could give me tag SNPs, that would be great. I would use them. If there was an easy way to take genotypes from the Illumina platform and give me the tag SNPs, that would be a good way of fitting the model.
So the question was asking Dr. De Los Campos to explain a little bit more about the reaction norm and how he used it in his analysis.
So in the standard reaction norm, the genetic value for the interaction is represented as the product of two scores. A score for the genetic value of the line and a score for the environment, the mean of the line and the mean of the environment, essentially. So what we did was to represent the genetic score as a linear combination of the markers, the environmental mean as a linear combination of environmental covariates, and then you take the product of the two. And what is relatively easy to show is that the covariance structure for that random effect is simply the cell-by-cell product of the G matrix times a sort of a quote-unquote G matrix computed with environmental covariates. So you compute the genomic relationship matrix, you compute the environmental relationship matrix, if you wish, and the interaction term has a covariance structure that is simply the cell-by-cell product of the two. So imagine that you have a G matrix with an entry that has a correlation of 0.5 between two lines. And then if these two lines were evaluated in two completely different environments, the environmental matrix will have a 0. When you take the product of 0.5 times 0, no covariance. So for the interaction terms, there is only covariance only provided that the two genotypes are similar and that they were evaluated in similar environmental conditions. Otherwise, there is no the reaction norm doesn't allow you to borrow information if there is no genetic similarity and environmental similarity. That's basically the idea.
In the genomic training analyses, we're typically not fitting any pedigree structure. So even if you know the pedigree, whether it's right or wrong, we're not using it. So it's not a problem. We're using the genotypes. There's still a problem that there can be a disconnect between the phenotype and the genotype. So, for instance, if an animal is measured live and then it's measured in a meat plant and then they take a DNA sample, we still have to be very careful that those always join up to the same animal, because there are times through that process where that can be broken up by accident, particularly when animals are detained on a processing chain. But provided the genotypes and the phenotypes come on the same animal, if we're out training on the phenotypic data itself, there's no problem. The problem is when we're, say, evaluating a bull and the bull wasn't measured for the trait because it's only measured in slaughter progeny or females, then we are relying on the pedigree to calculate the breeding value of that animal. And as long as the progeny test sizes are large, a few errors are not that important. But if there are lots of errors and they're, perhaps systematic in certain ways, then that could be a big problem. That's going to be less of a problem as time goes on, because there's a lot more use of small chips, which don't exactly resolve parentage, but they identify parentage errors. And so if we can find some causal mutations, I think we could see some small chips being used commercially for genomic prediction of commercial animals, and those same chips would have some markers for parentage. But having clean data is incredibly important. Of course, it makes the heritability larger and reduces the bias compared to if you've got data that has a lot of errors in it. And parentage is just one of them, but any step of the phenotypic process can have problems.
I think it's important to overemphasize that actually when we are talking about a lot here about using the molecular markers, using genomic to prediction, but actually if you don't have a good phenotype, you're not going to be able to do a good prediction. So that's what it was the point about. So I would like to intervene here and put the panel discussion to think about the next question. So the idea is that if you want to implement a genomic-wide prediction program now in the crops that you work with, that varies across the panel here, what will be the ideal population? Not only that, you have this contrast of putting a population that you have right now and try to maximize what you have right now, or actually construct a population that's the ideal to perform this kind of analysis. So,
well, I guess the approach I've been taking in the data breeding program is to mostly work with I think both approaches have been used. That you can construct. I understand. So you can design specific experiments. In any breeding program. Oh, you got it. Thank you. In any breeding program, you are always making crosses, and you're generating data which is potentially useful for these kinds of questions. Now, it's important, I guess, the distinction between what, if you just to take the normal data you would be doing in a normal breeding program, is that if you only ever, you have to always test your assumptions too. So it's important to carry through sometimes things that you didn't select, for example. If you always are validating based on selections that you've made based on a certain model, it limits sometimes your understanding of when the model didn't work. So I would say that initially, what we're going to do some cases where we advance things in the breeding program that ordinarily we wouldn't advance, because we want to compute some of these quantities like the accuracies. And once you measure accuracies for the different traits that you're interested in, and in the germplasm that you're working in, you can model the breeding program to design these parameters. And I tried to illustrate that with that one example. But questions like how many families should you have? How many families versus what size of a family? how many plots to use in this stage versus that stage. All those kinds of decisions can be, roughly speaking, can be determined based on economic analysis once you measure the accuracies, and those accuracies are the key parameters to measure. And the more you can measure those accuracies to represent a realistic scenario compared to what you would do in the breeding program, I think the better off you are in terms of designing a breeding program that's going to have the maximum genetic gain.
Yeah, I guess I'd just add to that you can always get faster gain by improving accuracy, and there are many ways to improve accuracy. Just collecting more phenotypic information is an obvious one. Genomics is just one method to buy accuracy. So when you're investing in genomics, you're just purchasing accuracy, and you can increase accuracy and increase your rate of genetic gain, but you don't always get a positive cost benefit from doing that. It depends entirely on the circumstances about how much the accuracy goes up, how much you can reduce the generation interval, and how much it costs. And at the current costs of genotyping, genomic prediction is not going to have a role equally across all different varieties and livestock species and plant species and so on. It all depends on what phenotypes you can collect at the time you make the selection decisions without genomics, and what the cost of those phenotypes are, and so on. So you have to do a conventional kind of breeding scheme design analysis to look at net present values and alternative schemes. And one of the difficulties of doing that right now with genomics is it's very hard to predict what the accuracy will be for each of the traits without actually having done the experiment to start with. So I think it's very important we're doing these in a number of species, and the value proposition will be incredibly good, at least in the livestock area, in species like dairy cattle, where you can't measure the bull for lactation performance himself, or say layer chickens, where they reach puberty a long time before you get the phenotypic information, and you can halve their generation interval. But there may be other species like sheep where you can collect many of the phenotypes at the time of normal selection, and in those cases the values of genomic prediction will be much less.
I think it's worth mentioning also a lot of these economic analyses you're going to do today, assuming a certain cost for genotyping, and those costs of genotyping might be only half a year later or a couple of years later, depending on the platform you're using. So that's a significant cost in adopting this kind of procedure that is changing quite rapidly. From our perspective, a lot of the folks that we talk to, especially in tree breeding, they don't necessarily yet want to embark in this kind of strategy without actually seeing the results from someone else. In other words, they're very reluctant in making a significant investment. What we think in those cases, it means that they won't be able to genotype as much as they should. And in that sense, to still obtain good accuracies, even if you're not genotyping at the scale perhaps that you would desire, working with a population that has an effective size that's relatively smaller is a good first step, because it allows them to do the genotyping at a lower cost, run their develop the prediction models, perhaps run a generation of breeding and tell whether if this is really suiting their expectations or not. So with that respect, I think the development of the population is really a critical side in terms of what kind of effective population size you're going to have.
I'm going to have two comments. One from the breeding perspective, I think, yes, ideally if you have a new tool like genomics, you will eventually change the structure of your breeding program. But I think that most of the experiences that people see around in the industry and applications try to do sort of a soft landing of genomic selection, where you just start to genotype the parents of the candidates of selection, you have the phenotypes and start to produce predictions, and this prediction can be used for selection decisions in the first step without changing the breeding system, because we need to learn a lot in terms of how genomic selection works before you change the breeding system. And the changing the structure of the breeding program, which is something that may have a lot of potential, is something that will come, I think, down the road after you have gained a lot of experience on using genomics with the current structure. As for the applications in humans, most of the interest is in predictions using unrelated individuals because family size is small and it's very difficult to imagine that you are going to have a lot of phenotypes and genotypes within families. Now the bad part is that these methods do not work very well with unrelated individuals, so there is a long way to go before this method can be applied for prediction with unrelated individuals for complex traits. If you have traits affected by a few loci, it can be done, but if you have complex traits, it's very difficult to do it, and there is a great need of research in terms of developing methods that can work in that particular context.
I would like to make a comment, and perhaps if some of the speakers want to complement, that especially in breeding, it goes down to the comparison of selection gain, and usually the benchmark is the phenotypic selection, which is what the breeder already does. But in most of the cases, I don't see the accuracies from the phenotypic selection being estimated through some sort of cross-validation. So I wonder whether this is really a fair comparison in the sense of benchmarking phenotypic selection compared to genomic selection coming out of a cross-validation scheme. And I want to hear from you what you guys think.
So usually we calculate the phenotypic breeding values or the traditional BLAP analysis based on these models that everybody was showing today. And actually those are based in infinitesimal models. So actually let's say that we are getting these breeding values based on an infinitesimal model. And then we are getting a trait that actually could have a simpler architecture. And then we are comparing and we are getting this accuracy of the correlation between these two things here. And actually you are getting a bad accuracy, and it might be driving actually because of one of them. The assumptions to get that phenotypic breeding value are wrong compared with the real structure of the trait. So please.
Numbers. Okay, so for instance, that's what in progeny, when people use data from progeny testing, that is not a big deal because every sire will have 3,000, 2,000, or 1,000 daughters in dairy cattle. So the estimated breeding value is a very good estimate. It doesn't depend on the assumptions of the model because the experimental design, the data dominates over the prior. It's the same thing that we're discussing. So you have large number of phenotypic records and you take average of those, even without any model, that is a very good estimate of the breeding value or the genetic value if you take average of phenotypes of an individual. So that's it. And the only true solution to the problem. Now, comparing the accuracy of individual phenotypic selection derived from a model versus a cross-validation accuracy, that's plain wrong. So it's just...
Yeah, I think I agree with Gustavo. I think we can look at, we have data to essentially try to assess this. I'm not really convinced that we will have lower accuracies if we follow cross-validation, but I think this is something we're interested in testing. Of course, you might have, in the cross-validation scheme, you're going to have a smaller population to define your parameters, so obviously there would be an expectation, I think, a loss of accuracy, but I'm not convinced that it's going to be significant.
Yeah, I'd reiterate Gustavo's comment. Once the technology is adopted and out there, the users, the producers will know very quickly whether we've done a good job or not. The problem is before that. If you run a small study and you have to be very careful because the sampling error of the validation correlation is huge. So in your little experiment, it could have looked a whole lot better, but then when you apply it validate it on a larger scale, it may not be as good as you thought it was. So that's something you have to be careful of.
Yeah, I'll just reiterate also that I would not. That's an apples and oranges comparison. You can't use the expected accuracies from the mixed model equations and compare it to an accuracy derived as a correlation between a prediction and a validation value. And at least in the comparisons I've done, the accuracies are computed on equal footing for both phenotypic and genomic selection.
Questions? I have time probably for a couple of rounds. Questions?
The technology is quite mature. Perhaps if we're considering Illumina genotyping or capture-based genotyping, the level of error is probably under 1%, and certainly very, very small. Now, there's a number of other approaches out there, like, for example, GBS-based approaches, where there is a tremendous amount of data that's missing, and where a lot of that data needs to be imputed. But there are some well-established ways by which you can clearly define your ability to impute the data properly or not. Simply, you might, take out some of the genotypic data that you know about, and actually then run your imputation and see how well you can recover that information. So at least from my perspective, I'm not as concerned about the genotypic error, because I know our expectation is probably is less than 1% for most of the platforms that we use commercially out there. I don't know.
Yeah, I would agree. The normal, say, Illumina SNP calls are pretty accurate, but I was peripherally involved in an experiment, a case control experiment, where the controls were 50k genotypes that had been taken on industry animals, and the case was done on a different platform, and it was actually another Illumina platform, and they got some incredibly good signals separating the individuals, but it turned out there was problems in clustering those few SNPs that came up with the strong signal. So after all the excitement had died down about finding incredibly predictive genotypes, a little bit more analysis showed that those were spurious. So you always have to be wary of those kind of things happening. But if your cases and controls or your whole population are all done at once, then the number of errors are relatively few. You still have to be careful with other kinds of issues. So for instance, some people test for Hardy-Weinberg equilibrium, but often the populations are from selected animals, and you wouldn't necessarily expect them to have to follow Hardy-Weinberg equilibrium. And I'm aware of one case where there was quite a large deletion, which turns out to have a huge effect on performance. And of course, anybody with the deletion appeared to be a homozygote. So those markers had been thrown out of the analyses because they didn't follow Hardy-Weinberg equilibrium, and they just thrown away the most exciting variant that they actually had in their population because they didn't behave the way they wanted to. So I think it's just like any science. You have to have your eyes open to the quality of data, and you have to have your eyes open in the interpretation of the results. And the cleaner your data can be, the better it can be, but you don't want to throw away good stuff.
Yeah, just echoing what Matthias said, I think for the array-based data, this isn't really the issue. It's more for methods like GBS, and maybe that's what you had in mind. In a number of the projects I've worked on at Cornell, we used GBS data because it was the place where that method was really pioneered. So we've done it in wheat, maize, cassava, and we spent a lot of time actually worrying about imputation. And maybe when I get to the software part, I can show you some of the options for imputation in the software that I developed. But the bottom line is that even for species like wheat, where we don't have a reference genome, there are. Predicting markers that are missing is another prediction problem, much like predicting phenotypes. And in fact, the random forest type of models, or random forest and its cousins, which is a non-parametric prediction model we have found to be very effective. So that's now the standard that we use for imputing missing GBS data when we don't have a reference genome. And if you do have a reference genome, then the hidden Markov models are very effective. And so I think it's, whether it's worth your effort or is I guess kind of depends upon the application. For whole genome prediction, sometimes actually you can show that the simplistic imputation is plenty good enough. Depends on the population size. If you're trying to do GWAS where you really need to know the marker data, then you have to do better than simply using the mean, for example. But if the goal is to estimate a covariance matrix between individuals, then the actual marker score at any given locus isn't really that critical. And so if that's the kind of analysis you're doing, then you can often get by with the simpler imputation approach.
Well, we've gone over the time a little bit. I'm going to ask a last question from the internet, and then we're going to wrap up with a last question from the audience here, and then break for lunch. The question from the internet was how to deal in a GWAS study with this idea of cross-validation, as when you select a training population, you're losing some power just by reducing the sample size. And then as an example, if you choose 5-fold cross-validation and in 3 of them the GWAS is significant, how to deal with that interpretation?
Yeah, I mean, we use cross-validation all the time to determine the accuracy of genomic prediction, but we don't routinely use cross-validation for GWAS. We've relied, in our circumstance, more on finding the same GWAS signal in a completely different data set, like, as I showed you before, in a different breed. That's much stronger evidence than you would get. I mean, it's hard to find a cross-validation data set within a breed that doesn't represent the same families, for instance. So you go to data from another company or another breed, and if you find the same thing, or else you validate through functional approaches, and you get into all the molecular techniques to do validation that way.
Just a very quick comment. Most of the times we use, I mean, you can do a cross-validation in a GWAS. Essentially the problem is the same than that of doing a cross-validation in a whole genome prediction. Typically we do cross-validation to estimate the distribution of the prediction errors and to get measures of accuracy. But actually we can use cross-validation as or resampling method for estimation purposes, and that's a very good example. So you have a marker that is highly significant in 3 out of the 5 folds. That's not the same as a marker that is highly significant in 5 out of the 5 folds. Because, and so there are some actually estimation methods that average over results of many samples from the population based on resampling, and those estimation methods are actually typically much better than the estimate that you will get from a single data analysis of the whole data set. So it's a whole thing about model averaging and estimation, but basically by averaging multiple estimates from resampling, you reduce sampling variability and you reduce mean square error. So actually there is a lot of information on that variability of the results across folds that can be used for estimation purposes as well. I think if your objective is to find causal mutations or to identify regions that affect the trait, that may be a good option. You may increase power. I think it has been shown that. Now, if your objective is to predict within a breeding program, the best thing that you can do is to genotype the ancestors of the candidates of selection, because you can exploit linkage, and that will give you the highest prediction accuracy. You're not going to get much accuracy by genotyping very extreme individuals that are not contributing to the breeding program. So these are two different things. For finding genes, I think it's probably a very good option. I don't have details about the specific experimental designs, but in regression, it is well known that if you have extremes, you have more power, provided that the model is true. But for prediction within a breeding program, it's a very different situation.
Probably also, if you have, it probably also depends on the type of trait you're working with. If you have a very complex architecture with many, many, many genes of very, very small effects, you're going to bulk these individuals into two groups, but actually within each one of these groups, actually the alleles in common that are, that favor a certain trait might be completely different, so you may not have a whole lot of power. But another reason why we call this in plants a bulk segment analysis. I don't know how this is called in cattle breeding. But the other limitation is that, of course, it constrains you to a single trait, right? If you want to look at different traits, it would be a different bulk. So there's been a tendency to sort of walk away a little bit, perhaps, unless you've got a high-value trait and a population is very suitable for that, for study of that given trait. And in those cases, sometimes it makes sense.
Yeah, I'd just reiterate the same comment. In my experience with it, we're normally interested in quite a number of traits that affect the breeding program, and you can take the tails for one trait, but by the time you take the tails of all of the traits, then you're doing an awful lot of the animals. The other issue is that once you do find something, you need to validate it, and so accounting appropriately for the selection that went into creating the tails means that you start needing to genotype some of the individuals in between. So it's much less of an issue now than it was a few years ago, because the genotyping costs have come down so much. It's easier just to genotype across a whole sample of individuals in most cases. But if you had a really high value trait and you wanted a first look, then it might be an appropriate way to do it, particularly in case controls. So it's quite often used in case controls, comparing, say, a rare disease where you only have the tail and taking a sample of the controls.
So with that, please join me to acknowledge the speakers. And thank you for coming, guys. And remember, we have lunch now in the hall out there, and around 1:45, probably 1:40, 1:45, we're going to be starting with the demonstration of the different software that the guest speakers have developed.